to augment those two. And the computer simulation is done because sometimes it's too difficult to build a physical experiment. Sometimes it's too expensive to conduct that experiment. Sometimes it's too slow to understand how things are going to evolve from the theory. Think of two galaxies colliding. That's an experiment that we really can't put together, but we can do a simulation of two galaxies colliding on a computer. And the same thing is true for climate. If we want to understand how the climate's going to evolve in the future, we could wait and see, but more importantly, we can conduct a simulation of that climate through a model that's been developed, and that will give us insight as to what's happening given the conditions that we have today. So computational science is a paradigm that augments theory and experimentation. It's considered this third pillar under which we do scientific discovery. And we use high-performance computing systems to do many of these simulation experiments. And those simulations are done on those supercomputers. So a supercomputer is today, a typical supercomputer is based on commodity processors. So we have a chip, maybe from Intel or AMD, and that chip has many processors on it. Those processors are called cores, and those cores sit on that one chip. They can carry out a computation using parallel processing, the ability to use those cores to help in solving one problem. And there's some memory associated with those cores as well. But we're not going to stop there. We're going to think about taking a board, and we're going to put multiple chips on the board. So we have chips now which have multiple cores, and we're going to put multiple chips on a board. So we have a large number of cores now on a board, again, with some memory. They're shared memory in the sense that each of those cores can get access to the memory quite easily. But those cores are limited in some sense in terms of their computational power. So we often think of augmenting those chips with graphical processing units, or GPUs. And those GPUs are there to boost the arithmetic intensity to help with getting more floating point operations in our computations. So we can offload things from that chip to the GPU. The GPU would carry out some computation and then send the results back to the chip. That's the way they're usually interconnected. And of course, we're not going to be satisfied with one board. We're going to think about having multiple boards in a cabinet connected through a network, so we can pass information from one process to another process in the course of doing a computation. That one cabinet is of course not going to be enough for carrying out the computations we're thinking about. So we think about filling up a room with a very large number of those cabinets. And you should think about the area of this, roughly the size of 2 tennis courts would be the right approximate size. And we have a number of supercomputers which have been designed and built and are in operation today. There are some Chinese supercomputers. There's a supercomputer in the US, which is considered the fastest supercomputer, built exactly on these principles of taking commodity processors, putting them on a board, augmenting that with an accelerator in the form of a graphical processing unit, and putting many of them together in cabinets and connecting them together to form a single unit, which we do parallel processing on to effectively compute the solution. So today's environment for scientific computing is really highly parallel. It uses distributed memory. That is, each of the nodes of this computer has its own memory. And in order for a node to share information with another node, it has to engage in a message passing paradigm. And for message passing, we use a programming model called MPI, the message passing interface. And we also use something called OpenMP as the programming paradigm when we share information on one of the nodes itself. The fastest computer is this computer at Oak Ridge National Laboratory. It's a Department of Energy laboratory and it has roughly 8 million cores in it. So 8 million cores, 9,408 nodes in this computer. They use parts from the company AMD. AMD CPUs and AMD GPUs are in this computer, the supercomputer. And if we take a look at the where the performance is, 98% of the performance comes from the GPUs. So you really need to be using those GPUs in your computation, that offload of the computationally intense parts, and doing that on the GPU. So we have this heterogeneous collection of commodity processors and also commodity GPUs that are being used to drive high performance computing today. The thing that's an expensive part, I'll say, of doing a computation is in communication. So we have to pass information from one process to another, and that passing of information is done through messages. It's done through MPI, and it turns out that that particular thing, communicating, is very expensive on the supercomputers when compared to the floating point operations. So floating point operations are relatively cheap, and message passing communication is relatively more expensive. So that's a change in some sense in how we used to think about computing. We used to think about computing in terms of the floating point being the most expensive part, and today that's changed quite a bit. And the other thing to realize is that we have different levels that we can use to do our computations. Much of scientific computing in the past has been done using 64 bits of precision. So we have a word, and that word has 64 bits in it, and we multiply and add operands which have 64 bits of precision, getting a result which is 64-bit accurate. And we can think about 32-bit, that's considered short precision or half precision. And then more recently, we have 16-bit and 8-bit floating point arithmetic. The 16 and 8-bit floating point arithmetic has been provided to help applications that use machine learning. Machine learning, it turns out, can get away with short precision. It doesn't need all of those bits of accuracy, and the hardware vendors have adjusted their computing power so that it does that short bit of arithmetic at much faster rates than it can using the 64-bit or 32-bit. So today we see many architectures with 64-bit, 32-bit, 16-bit, and even 8-bit floating point operations in them. The fastest computers today are considered computers that are running at an exaflop. An exaflop is, when we're talking about floating point operations, we're talking about an add or a multiplication of a 64-bit operand. So we will be using 64 bits when we describe the performance here. So one operation, one flop, is an add of two operands, both having 64 bits, or one flop is a multiplication where we take two numbers, 64-bit numbers, and multiply them together. So an exaflop is a billion floating point operations. So that's 10 to the 18 floating point operations can be completed every second on this on the fastest computers, and that fastest computer would be called an exaflop computer at that point. So that's where we are in terms of the state of the art. To think about an exaflop, to think about a 10 to the 18 operations. If each person on Earth were to do one calculation a second, it would take more than 4 years to do a billion operations, or 10 to the 18 operations. So 4 years for everybody to computing on Earth to do what this computer can do in 1 second, just to put that in contrast or relief. So it really is quite an astounding amount of computation. I became a benchmarker back in 1979. We were developing a project called Linpak. Linpak was a project developed by the National Science Foundation. It was funded by the National Science Foundation and the Department of Energy. And it involved 4 groups. It involved, in this picture here, the person on the right is Jim Bunch. He was from the University of California, San Diego. That's Pete Stewart from the University of Maryland, Cleve Moeller from the University of New Mexico, and that's the 1979 version of me with a little bit more hair. That's my car that we're leaning on. That's the license plate that I had when I was a student at the University of New Mexico, and we worked on the Linpak project during that time. We produced a user's guide for Linpak, and in the appendix to the user's guide, I put together a little table that listed the performance of about 20-some machines. It listed the performance, how fast they were able to solve a system of linear equations. That linear equation was of size 100. So think about a matrix problem, AX equals B, and think about solving that using a very standard algorithm, Gaussian elimination with partial pivoting. The Linpak user's guide has that routine in it, and I put together a benchmark that solved that system of equations. So this is a table this is the table that's in the appendix of that user's guide. It shows 23 machines. It lists the performance or the time it took to solve a system of equations of size 100. The fastest computer was a machine called the Cray 1 that was at the National Center for Atmospheric Research in Boulder, Colorado, and it ran at a rate of 14 megaflops, 14 million floating point operations per second. So that was back in 1977. A lot has changed since then. A number of years later, I had a list. That list of 23 machines grew to be a list of a few thousand machines. I had a colleague in Germany, Hans Moyer and Erich Strohmeyer, who had a list of the fastest computers, and they suggested we put together our list of their fast machines and my benchmark results, and we call it the top 500. So that's the current thing that we use to rank supercomputers, is called the top 500. It's based on solving a system of linear equations. That system of linear equations then gives you a ranking in terms of the fastest computers. We only list the 500, the fastest computers in this list. The list comes out twice a year. And again, we're solving AX equals B. The ground rules say you must use Gaussian elimination with partial pivoting. I'm going to give you a matrix. You have to solve that matrix problem. I'm going to give you a matrix A and a right-hand side B, and you have to find X using that algorithm. And you can implement the algorithm as fast or using the best techniques as you can. And then we're going to look at the rate of execution to compute this flops per second rate, and that's going to be used as an entry point. So the list comes out twice a year. It comes out in November and also in June. The most recent list is from November of 2022, and it shows the rankings then for these computers. So this is, in some sense, a snapshot of high performance computing going back to 1993 when we started. The red dots indicate the fastest computer. So today, the #1 computer is at 1.1 exaflops. The computer that's on the bottom of the list today, that's at 1.73 petaflops, or 1.73 times 10 to the 12 floating point operations per second. So that's the bracketing, if you will, for high performance computing. And we can see a lot of trends and so on. So the blue line is the sum of the 500 computers. That's an artificial Excel sheet computation. But back in 1993, the fastest computer was a machine at Los Alamos National Lab. It was used for nuclear weapons design. It was a computer called the Thinking Machine CM5, and it had 1,000 processors. I'm giving a talk on a laptop, and my laptop has the ability to solve that benchmark problem. And when I solve that benchmark problem, I get 426 gigaflops. So that my laptop is really faster than that #1 machine back in 1993. In fact, it's equivalent to a computer that was #1 in 1997. It was a Hitachi machine with 2,000 processors at the University of Tsukuba that was used to do many numerical computations that were of importance to the people in Japan. So a lot has changed in a very short period of time, and that change is continuing today. So this is a list of the top 10 supercomputers in the world. So the number one computer is this computer at Oak Ridge National Lab, as I mentioned. It's called Frontier. It's integrated by HPE. That's a computer company. They don't make chips, they integrate things. So they take AMD parts, they take AMD processors and AMD GPUs, and they put them together with an interconnect called Slingshot from a company called Cray, and they make a supercomputer which they sell. And that supercomputer has 7.7 million cores or processors in it. It reached 1.1 exaflops, and that's 65% of the theoretical peak performance. So think of the theoretical peak as a paper and pencil computation by looking at the cycle time of the machines, of the processors, looking at how many operations you could do per cycle, and then computing the execution rate if you did nothing but those floating point operations. So when you run the benchmark, you get 65% of that peak performance, let's call it. And you could see how that varies with other computers on the list. The computer these supercomputers are very, they need a lot of power. So the Frontier machine needs 21 megawatts of power to run. So this benchmark, when running, you consume 21 megawatts of power. Think of it like this: a megawatt here in Tennessee, where I live, if I use 1 megawatt for 1 year, I would get a bill from the electric company for about $1 million. So that's a megawatt year, $1 million. And so this computer, this Frontier computer, consuming 21 megawatts, that's going to be running for many, many years. Every year, the power bill for the computer is roughly $20 million, $20 million US dollars. So you could see these are very expensive machines to run. The computer itself cost about $600 million, and it's very, very powerful. So when we look at the top 10 list here, we see the US has five computers. There are 2 computers in China. So China has some computers that they're designing on their own. They're not done by Western companies. This first one here, the Sunway Tahu Light, is actually using a Chinese chip from Sunway. It's a chip which has 260 cores on it, and it's number 7 on the list. There's another machine that's number 10, the Tianhe 2A, and that machine is using Intel parts. While a number of years ago, China was still allowed to use Intel parts, today there's an embargo against China from using parts from the US companies. So they're changing this architecture today to replace these Intel parts with processors that are designed in China. They're going to be using ARM. They're going to be using an ARM processor to drive this computer in the future. So this gives you a sense of the computers here. The red indicates that they have accelerators or GPUs. The one that doesn't have a GPU is this machine from Japan called Fugaku. It's based on an ARM architecture, which uses vector instructions to help boost its performance. And you can see the performance rate. They actually do very good in terms of their theoretical peak. They're running at 82% of the theoretical peak of the machine. So 5 in the US, 2 in China, 1 in Japan, 1 in Finland, and 1 in Italy for the top 10 machines. These are very powerful systems being used to do advanced computing. The number 1 system, I'll just want to talk about it just briefly here. It has the peak performance of 2 exaflops. So 2 times 10 to the 18 floating point operations per second. Each node of this computer has 1 AMD CPU and 4 AMD GPUs in it. And it has 9,408 nodes in the machine. And the interesting fact is that if we take a look at of the performance, if we use short precision, 16-bit floating point, instead of 2 exaflops, that's for 64-bit floating point, at 16-bit operations, the performance jumps to a peak of 11 exaflops. So, there's a factor of 5 there in terms of the boost given by using that short precision in it. We also noticed that most of the performance, 99% of the performance from this computer comes from using those GPUs. So you really need to be using that component, that GPU in these systems in order to be getting the fastest execution rate. This is looking at the #1 computer over time based on the top 500 data. You could see how it's changed. We reached what was called the teraflop, 10 to the 12 operations back in 1997. 11 years later, we got 3 orders of magnitude in getting to a petaflop, and then it took us 14 years to get to an exaflop from that point. So 3 orders of magnitude going from tera to peta in the 11 years, and then going from peta to exa, it took us 14 years, and we would expect carrying on into the future, it's going to take even longer. So things are slowing down, and that slowdown is coming about by a number of things. Some of it driven by Moore's law, other things driven by something called Dennard scaling, and it's just harder to get the performance. So if we take a look at a projection here, the blue is giving us an indication of where the #1 computer is today. It's just over 1 exaflop, and we see that we're going to reach an order of magnitude greater performance, 10 exaflops, based on this projection here in 8 years. So it's going to take us a long time to get 3 orders of magnitude to reach the next large threshold, which would be zettaflop computing. So we'll have to see how that evolves over time. If we take a look at the supercomputers and look at where they're located, China has the most supercomputers. So there's 162 of the 500 fastest computers. China has 162. US has 125. Germany, Japan, France, the UK, et cetera, have those numbers associated with it. China also produces the most supercomputers. So not only do they have the most supercomputers in their country, they make the most supercomputers, which they then sell worldwide. So they have 5 manufacturers in China: Lenovo, Sugan, Inspur, Huawei, and the National University for Defense Technology has been designing and building supercomputers for many years and has been deploying them. There are also rumored to be 2 exascale computers in China, but those computers have not run the benchmark, or they haven't submitted the benchmark so that they're listed in the top 500 list. So the first one is a machine called the Sunway Ocean Light. That's a follow-on to the previous machine that is listed in the top 500. And it's reputed to have over an exaflop for Linpak. There's been scientific papers written about this computer, so we understand the architecture very well. There's also another machine in Tianjin in China called the Tianhe 3, and that uses ARM processors and an accelerator. And also it is reported to have performance above an exaflop for the benchmark, but they haven't submitted the results. And I think there's concern in China if they were to submit results, it would probably cause more issues with computer parts going to China. So I think they're just lying low and not letting these machines be widely known that they exist. Supercomputers are used in industry. Industry provides the ability to develop new and better products using high-performance computers. We see it being used in many areas. In fact, if you take a look at the top 500 list, about half of the machines, half of the computers are used in industry. So industry understands that these computers provide some strategic advantage for them in getting things, making better products. And they're being used in many fields. So as an example, they're being used in the oil industry for seismic data, trying to locate oil reservoirs or other things under the ground. They send a pulse on the ground. They look at the pulse and they try to reverse engineer what's under the ground based on the signals they get back from that. Aerospace industry uses high-performance computers to design better aircrafts, make them better structural, make them more efficient. The automobile industry uses supercomputers to help design lighter, more economical, more crash-worthy vehicles. In the old days, they would build something and crash it into a wall and see what happens. They probably do a few of those experiments, but most of the experiments are done using computer simulation on high-performance computers to be able to optimize the design of that product. And the chemical and pharmaceutical industry uses high-performance computers to design better drugs to combat certain diseases, certain illnesses as well. Again, if we take a look at the market segment, now we're looking at the top 500 from the beginning in 1993 until today, and we're looking at a breakdown of where they're being used. We see that industry has 233 machines today are being used in industry. 119 of those machines are being used in research, and down the list you could see that. If we take a look at the machines in China, 134 of their machines are being used for industry purposes. In the US, 39 of the US machines are being used as an indicator for industry. So high-performance computing is being used for simulation. It's being used to carry out simulation for climate, combustion, nuclear reactors, fusion, stockpiled stewardship, material design. It's being used in accelerators. And most of these problems are being simulated through the use of three-dimensional partial differential equations. So we use 3D partial differential equations to do the modeling, and those models result in a discretization, and that discretization leads to solving a system of equations. Very similar to the Linpak benchmark, we want to solve AX equals B, but here the matrix is not dense, it's sparse. It has a sparse structure associated with it, so there's not a dense structure. And that algorithm we used for the Linpak benchmark, Gaussian elimination with partial pivoting, doesn't work very well here. Because Gaussian elimination expects the matrix to have a lot of non-zeros, and these matrices that are used for partial differential equations are relatively sparse in nature. We use a different algorithm. We use an algorithm which is iterative in nature, and we use a method that we call the conjugate gradient method for solving it. So again, we're solving AX equals B. A is large and sparse. We have a right-hand side B, and we want to compute X. So we use an iterative process to do that, and we have a matrix which has this structure associated with it. It's sparse. It has that sort of non-sparse, non-zero structure associated with it. We have a discretization. In this case, it's a 27-point stencil operator that we use to discretize that 3D partial differential equation, and that leads to this matrix problem. So we have a benchmark now which uses this algorithm, the HPCG. So this is something that's more like the real method that are used on computers to solve 3D partial differential equations. So here's a list of the top 10 machines for doing HPCG. I've listed here the Linpak number. HPL is the Linpak number for the machine. The number one system for solving HPCG is the machine in Japan, and it had a Linpak number of 442 petaflops, and it has a number, the rate that it solved this benchmark is 16 petaflops. So you see the difference there between solving a dense matrix problem as opposed to solving a problem which is based on a sparse structure associated with it. And I've listed here the percent of peak performance. So in the best case, we're getting 3% of the peak performance of this computer. And if we take a look at the exascale computer, the machine at Oak Ridge, in this case, we're getting just 0.8% of the peak performance of the machine. So less than 1% of the peak performance. So that's a warning in some sense that that high level of performance is something which is going to be very hard to obtain on this computer. So this is a graph here which gives us an indication of the top 500. The black dots are the theoretical peak performance. The green dots are the Linpak performance numbers. And if we look at the HPCG numbers, they're much less. And that shows us a bracketing, if you will, from the blue is really the real applications, and the green is where we are with the benchmark that was designed in the late 70s, where floating point arithmetic was very expensive. Today, floating point arithmetic is not expensive, and the communication is really the expensive part. So this new benchmark reflects better on the communication. So if we take a look at the top 500 machines and how they've changed over the years, what we see is that Intel has been supplying parts for supercomputers for a very long time. But more recently, Intel and AMD, both of which use something called the x86 instruction set. Both of them really dominate the top 500 list. So we see, 97, 98% of the top 500 machines are using Intel commodity processors, or AMD commodity processors, which are based on x86 architecture. Only 5 computers on the top 500 list are using ARM processors. ARM is probably the most common processor. It's used on our cell phones. And at some point, we would expect the ARM architecture to also become a more dominant player in this way. The other thing that we have is that we have accelerators coming from really just a few companies. Nvidia really dominates the accelerator market. So those GPUs are coming from Nvidia. The interconnect for our high performance computers are really based on Ethernet or InfiniBand. They dominate the internet, the interconnects on the supercomputers, and we see Linux everywhere. So we have commodity processors, commodity interconnect, commodity GPUs being used on our systems, and that, of course, is driven by cost. So we want to buy a low-cost machine which has high performance. And if we could buy something off the shelf rather than to design it ourselves, we would be in a better position for the cost. When we look at high performance computing, we typically see a small amount of data that comes in, and then we have a numerically intensive computation that takes place, and a large amount of data that comes out. So that's what we would consider more a traditional high-performance computing application that's very numerically intensive. Here's an example of that. We're looking at the climate model as it evolves over time. We have a small amount of data that goes in, and we get this very rich view of what's happened over time as output from that. And that's sort of in contrast with the high-performance data analytic solutions that we have. They have a large amount of data going in. That data going in is used basically to train the models, and then we have a small amount of data coming out, the answer that we're looking for from machine learning. So a large amount of data going into machine learning and a small amount of data coming out. Quite different than what we see for scientific computing. Usually we're thinking about 64-bit input. The simulation is done in 64-bit, and then a solution comes out, which is represented as 64 bits of data. With machine learning, there's 16-bit floating point operations as input to the learning process and inference process, and then the solution comes out as, again, just a 16-bit output. So it's quite different in terms of the numerically intensive and the data intensive applications that we have. So machine learning and artificial intelligence have been around for a long time, but they've really taken off quite recently. And the question is, why did that happen now? Well, we have this flood of data, especially with the internet, to help train the models that we have. So we're pulling data from all over the internet to train specific components of these models. We have this increasing computational power that comes from these GPUs that we use. We have we have progress that's been made in terms of the algorithms and the theory behind the artificial intelligence and machine learning that have been developed over the last few years. And we have this increasing support from industry that's embracing machine learning and using it to help them drive what they're doing. And that also happens, of course, in scientific computing as well. We see machine learning being used in many areas. And we see a lot of hardware that's being designed specifically to help with machine learning and artificial intelligence. So these are some of the companies that have produced hardware that are really intended for use within the machine learning process. Companies like Cerebras and Sambanova and GraphCore all have tools that can be used to help boost the performance. You know, artificial intelligence is a large area, and we can think of breaking it out into smaller groups. There's one component that we think about machine learning that helps us. There's also natural language processing. If you've used ChatGPT, that would fit into this niche here. Expert systems are another form of artificial intelligence, vision recognition, speech processing, speech to text and text to speech, and robotics and planning all are part of the artificial intelligence revolution. So machine learning is important. It's a vital part of computational science. It's a tool that computational scientists are using, and we see it being used in many areas: climate modeling, biology, drug design, materials, high energy physics are all using machine learning today to help drive their understanding. So today we see supercomputers being built with commodity CPUs and GPUs. And the question might be, what happens in the future? Well, I predict that in the future we're going to have many more things added to that ensemble. We'll have CPUs, GPUs, machine learning devices. We'll have neuromorphic computing, maybe quantum computing as an add-on to this ensemble. Tensors and optical computing all have a place that could fit into this as we go forward into the computing, into looking at better ways of doing our computations. And we might be able to dial up, the number of these components that we have on our supercomputers to give us the right balance for the applications that we want to solve. So in conclusion, hardware is in constant state of change. We go from scalar to vector to distributed computing to accelerated, and we have now mixed precision. There are these three computer revolutions that have taken place. High performance computing is one, deep learning is another, and edge computing, which I really haven't had a chance to talk about, is yet a third level. Algorithms and software are critical for all of this. And the advances that we see in the algorithms and software really follow from the hardware. So the notion I have is that supercomputers are designed, the hardware is designed, and it's thrown over the fence, and it's left up to the applications people to look at the design of algorithms and the implementation of that in software to fit onto the hardware that they've been given. And sometimes it takes a long time to get an optimal match between the hardware and software. And we're not going to be doing that very efficiently initially, and we'll have to refine that over time. There was an interesting paper written by Charles Lyserson that appeared in Science in 2020, which described this. In fact, the title of their paper was "There's Plenty of Room at the Top. What will drive computer performance after Moore's Law?" It's a riff on Feynman's 1959 lecture about there's plenty of room at the bottom, where he was talking about quantum computing and looking at how things could be designed at that level. So that's sort of where we are today. There's plenty of opportunity here to develop software and algorithms for high performance computing that fits with hardware development. So thank you so much for your attention, and I guess we'll answer some questions in the follow-up. So bye for now.
Thank you, Prof. Dongarra. Our next speaker is Prof Robert Langer. Prof Langer is joining us virtually and will share about the topic from nanotechnology to mRNA therapies and what's to come. How overcoming skepticism and barriers led to new vaccines and ways to tackle global health challenges. The Q&A session will be moderated by Prof Ali Mizrahi. Prof Langer, please.
So why don't we, let me go over how I got started. So I was a chemical engineer, and all my colleagues would go to chemical engineering jobs like, thank you for the slides. They, 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 they would go to oil companies. So I thought I would do that too. So I wrote, so a lot of the oil companies would come to MIT, and I actually got 20 job offers from oil companies, a lot, even about 4 or 5 from Exxon alone. And I wasn't that good, but they just had so many openings. So one of the, but I felt when I went to these companies that the impact I'd make wasn't that great. You know, they'd say you could spend your life improving the yield of some of these chemicals by 0.01%, and that would be what you do. And I thought I didn't want to spend my life doing that. And so I started thinking of other jobs I might do that could help people. And I was very interested in education. I helped start a school for poor high school kids. And I got very involved in creating new chemistry curricula. So I wrote, one day I saw an ad at one of the universities to do that. And I wrote them applying for a job, but they didn't write me back. But I tried about 40 different colleges to do that, and none of them wrote me back. So that wasn't going so well. So I thought other ways I might help people were by using my chemistry and engineering background in medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. But then one day I walked into the lab, and one of the older scientists said to me that, Bob, you know, there's a surgeon in Boston named Judah Folkman, and sometimes he hires unusual people. Um, he thought very highly of Dr. Folkman. I won't say what he thought about me, but I wrote Dr. Folkman a letter, and he offered me this job. And he had a very unusual idea. This was 1974. And if we could go to the next slide. Could So this was his idea. His idea, this is a picture of how a tumor grows. If you go on the lower left, the idea is the tumor is somehow abnormal. It grows as a three-dimensional mass, and and if you go to the center, you can see that the center bottom, it really doesn't grow beyond about 1 million cells. So, but the way the tumor solves this issue about getting bigger, see, it's a nutrition problem. Cells in the center can't get nutrients. So what he suggested was tumors make substances called TAF, or tumor angiogenesis factor, and that causes the surrounding blood vessels to grow to the tumor. And then when you go to the upper right, notice how now the tumor gets bigger and bigger. And on the utmost right, it's very vascularized, and the tumor can spread through those blood vessels. That's a process called metastasis, and that often kills people. So this was his idea, that this is how tumors grew, and most people didn't believe him. I didn't know anything about biology, so I thought it was a great idea. Uh, but there was no way to study this. So we had to develop what's called a bioassay. And there was no way to study blood vessels then. All these molecules that we were isolating to try to see if they could stop blood vessels, they were big molecules, and we had to deliver them for a period of time. And no one had done that before. And Dr. Folkman, he was on a board of the only company in the world, Alza, that was working on this. And so we went and asked them, could they help? But if we go to the next slide, what they said is that you could never do that. this is just, this was the assay we wanted to use, that you'd put a tumor in the eye, and then you'd have a tiny nanoparticle or microparticle delivery system that would deliver these large molecules that I was mentioning. And like I say, he asked these people that were real experts in this, could they ever help us? But they all said, next slide, that they couldn't, because the molecules were too big. If we could go to the next slide. Yeah, they just said they were too big to get out of these, through these solid materials. So anyhow, if we were going to solve this problem of blood vessels, I had to solve the problem of delivery. So I spent 2 years in the laboratory experimenting with different materials and different designs. I found over 200 different ways to get this to not work. But eventually, if we could go to the next slide, I was able to make tiny little micro or nanoparticles. You see a picture of one on the left, cut in half on the right. And what we showed on the next slide is that you could use this approach to deliver molecules of any size. This was a paper in Nature we wrote in 1976. So you could release molecules of any size, even nucleic acids like mRNA or DNA for the very first time. But when I first presented this work, it was really ridiculed. I was 27 years old, probably like the age of some of you. And I was the youngest person at this scientific meeting of chemists. And I gave this talk. And when I got done, I thought I didn't do that badly because I didn't forget too much what I wanted to say. So I thought all these older people, being, nice people, they would want to encourage me, this young guy. But when I got done, I stepped off the podium and a whole bunch of them came up to me and they said, "We don't believe anything you just said." If we could go to the next slide. Again, they said that the large molecules were too big to diffuse through little cages of like nano or microparticles. And also they were pointing out that and this is true, I used organic solvents like ethanol and methylene chloride to do these encapsulations. And they said that would destroy all these molecules I put in. So that wasn't so good. And actually things kind of went downhill for me from there, if that were possible. I wrote grants to try to fund some of our work, and my first 9 were rejected very, very soundly. I also, I really liked being a postdoc, but a lot of my friends said, I should apply for faculty positions. And like I said, I was a chemical engineer. So I did. I applied for faculty positions. No chemical engineering department in the world would hire me. They all said, this bio stuff you're doing doesn't make any sense for a chemical engineer. You should be doing oil or energy. So that wasn't so good either. Finally, I got a job in the nutrition department. But the problem there was the department head, he was what I'll call a benevolent dictator kind of department head. He liked me, so he offered me a job, but he didn't bother to ask anybody else in the department what they thought. Now, that might have been okay, except for one thing, which was the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. If we could go to the next slide. My friend Mike Marlette, he's a very famous chemist who was head of Berkeley's chemistry department. He was reflecting a few years ago when I got some award, what it was like to be with me in 1979 at MIT. And here's what he said. One evening, I went to a faculty dinner at a Chinese restaurant with myself and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. So I had a very inauspicious beginning, but I really believed in what I was doing. I could see a lot of this with my own eyes, that we put different dyes in and they of large molecules and they would come out. And so we kept at it. And I mentioned to you that one of the problems we wanted to solve was the blood vessel problem. So I also, at the same time, was isolating different substances, and I had different fractions, and so I would put them in these little nano or microparticles, and we had that rabbit eye assay. And almost every one we isolated did not work. We did about 2,000 eyes, but one did. And let me just show you, and I apologize for showing such a bloody slide, but let me just show you on the next slide what happens. So if we put, these were the cornea, if after a period of time you put a sham or inactive fraction, you get what you see on the left, say after a couple of weeks. Ultimately, those tumors would kill, but we sacrificed the animals before. But when you put this one inhibitor in, look on the right, and we repeated this 20 times, the vessels are lower, they're sparser, and they avoid the tumor. We published this in Science in 1976, and that really, I like to think, began to change people's thinking. Nonetheless, it still took 28 years from this Science paper before the first blood vessel inhibitor was approved by FDA. And that took great work from Genentech and other companies. But the next slide was kind of striking about what ultimately happened. And this may be a little hard to see, but what you see is that starting in 2004, 28 years after our paper, one after the next blood vessel inhibitor is approved by regulatory authorities. Avastin, the first of these, is one of the 2 or 3 largest selling drugs, biotech drugs in history. And not only can you treat all kinds of cancer this way, as you see in the right, but eye diseases like macular degeneration, one of the leading causes of blindness in the world due to abnormal vascularization, there was no pharmacologic treatment before. Now drugs like Eylea and Lucentis are blockbusters, treating many millions of patients with these diseases. So, like I say, this would lead to all these inhibitors. But I also thought that the drug delivery work itself would be useful. So one of the things Dr. Folkman said to me one day is, Bob, we should file a patent. And Children's Hospital had never had a patent before. I didn't either. So we filed the patent. 5 years in a row, the patent examiner rejected it. And the people at the hospital came to see me and they said, Bob, you should just give up. This guy's never going to allow it. But you can tell by now, I don't like to give up. So I started to think, how could we convince the patent examiner to allow this, legally, of course. And science really wasn't working. But as I mentioned to you in the beginning, that when we first started doing this, everybody told me this was impossible. It could never work. I wondered, maybe someone wrote that down. So now in 1982, I did a science citation search to see who cited our 1976 Nature paper. And I found it's always interesting to see what people say about you. At any rate, let me just show you one article. Next slide. So I had no idea this was written, but it was 5 of the top chemists and engineers in the world in 1979. I'll just read you what they wrote, describing this really embryonic field. They said, generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising, I thought surprising, that's a really good word for a patent examiner, results that clearly demonstrate the opposite. So I showed this to the people at the hospital, they showed it to the patent examiner, and he said, gee, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from those 5 people that they really wrote it. So I wrote them, and they were all nice enough to write me back they really wrote it, and the examiner did allow this patent that you can see on this slide. Now, with that patent, we actually licensed it, and I consulted for a couple of very large multi-billion-dollar companies. I was so excited. They gave me grants, and they were, giant companies. And I thought, gee, something I did might help people someday. But, they worked on it maybe for a year, and they just gave up. And then one of my colleagues, Alex Klevenoff, said to me one day, Bob, said, we should start our own company. So we started this little company, Enzatech, which later merged to become Alchemies. And we also started other companies and also licensed things to different companies. And that has led to many, many products, some of which are shown on the next slide. And there are all kinds of things that you can inject into the body that can treat cancer, heart disease, schizophrenia, type 2 diabetes, even like what are called drug-eluting stents, which is in the bottom right, which is one of the major treatments for heart disease. So hundreds of millions of people now use these every year. But going back to the academic lab, I could still see that there were lots of issues to solve. When we made most of these that you're just seeing here are like microspheres or implants. As we make things smaller and smaller, you could get release like we showed in that Nature paper, but sometimes what would happen is the little particles, like the nanoparticles, they'd stick together and aggregate. And also they could get eaten by macrophages. So we had this idea, next slide, of making a special coating. We did this with Vladimir Torchelin and others, and called polyethylene glycol. This will end up being one of the 4 key components in all lipid nanoparticles. But now, this is almost 30 years ago. So we published this in Science. And let me just show you a little bit of data. The next slide shows some atomic force micrographs. The next slide just shows you that they really are nanoparticles. Next slide. The next slide is quasi-elastic light scattering. And what's really exciting is shown on the next slide, that when you put an orange dye in them and you have a macrophage, if you don't have PEG, look on the upper left. They're all eaten. They're all in the cell within an hour. But when we coat them with PEG, they're hardly taken up at all. And there are different designs we use. So that was very exciting. And then we thought, well, we could even do targeting. And the next slide is sort of that idea that, and Omid Farrakhanazad did a lot of that. And by the way, sometimes I feel I don't explain this that well. So I want to show you a video on the next slide that Nova, the TV show, did. If we could turn on that next slide in video.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
Now, I should tell you that all the clinicians I work with tell me it doesn't blow the cell up quite like that. But at any rate, so that's what we thought about. And then we also, with Dan Anderson, who was one of my postdocs, decided to do work on the 4th component. Like there's 4 components in lipid nanoparticles: cholesterol, structural lipid, now a PEG lipid, because in part of what I just showed you, and then like what's called an ionizable lipid. But when we started, there were very few lipids that people used. So we came up really with the first ways of doing what I'll call combinatorial synthesis of lipids, where we could make thousands of them. And this was the wish list shown here, that you could do it in a single step. And part of what we did is use certain types of chemistry, illustrated on the next slide, like a Michel addition. And by doing that, you could react diamines, and basically you could make lipid-like materials. And you could get enormous chemical diversity by changing the R group. And the next slide or two will show you that. The next slide just shows large variation in the R group. And you can also change the tails and number of tails, as shown in the next slide. And so you get enormous diversity. So we literally made thousands of tiny little lipid nanoparticles. And on the next slide, this is all work we did in collaboration with El Nilam, with Akina Kink, who was one of my students, but then went there to lead their program on rare diseases for ATTR amyloidosis. So you could get a dose response curve. You could get this is factor 7 in vivo, but the next slide shows you get a dose response curve. As you increase the amounts, you get more and more knockdown. The next slide just shows you can get a real treatment possibly for atherosclerosis by knocking down ApoB with some of these. And you can see that here. And you can give these, we find you can give these siRNAs repeatedly. You don't have to just you can give it once and it might last for a month. But the next slide just shows you can keep doing it over and over, which was exciting. And then, of course, Dan and I, you want to do better. So what we did is we could take the initial libraries we had, but then we could improve them. And an example of that shown on the next slide, that we could take the amines from the original library, take the best ones, but now use an epoxide chemistry to make 150 new compounds that we could test in vitro and then in vivo. And let me just show you the data as we improved the efficacy by this 4th lipid. So what you're seeing on the left is the LNP01. That's what I showed you earlier when you get knocked down. But you can go maybe almost 2 orders of magnitude better by doing the epoxide chemistry one I just showed you. And that's in the middle. So you get this dose response curve, but you get some knockdown with as low as 0.003 mg per kg. And that translates to monkeys, next slide, non-human primates. And again, you can get this with not only, with non-human primates, very significant knockdown. And over a 10-year period, our cells working back and forth with Alnylam and some of their collaborators in Canada, we'd go back and forth, improving the potency. And the next slide just shows you what it shows you. It started out with, in 2004, you could see, if you go to the right-hand side of the screen, you get these first generation nanoparticles, but you can keep improving them and improving them. And by 10 years later, you're up to about 4 orders of magnitude better, more potent. So that was very exciting. And I mentioned Kinnikinck, and he led this effort. He was my student at Alnylam. And what that would ultimately lead to is shown on the next slide, which is Onpattro. This was the first lipid nanoparticle with a nucleic acid, or first nanoparticle with a nucleic acid in it. It's used to treat a rare disease, ATTR amyloidosis. It's a nerve disease, and it's been really a wonderful thing for those patients. So at any rate, that was 2018. Now, of course, we started a lot of the work a lot before that, but even back in the 70s. But in 2010, now go back in time, myself, Derek Rossi, Nubar Fayed, and Ken Chen, we had this idea that, well, okay, siRNA, that knocks genes down, but messenger RNA, you could get the opposite effect. You could get gain of function. The next slide is really just a central dogma of molecular biology, that DNA makes RNA makes proteins. Well, by this time, 2010, there were enormous number of protein therapeutics that had come about in the last 30 years. But I'm sure you all know it takes a long time to manufacture them. You need these giant cell factories. In the case of some vaccines, you need eggs. It may take a year to do it. Here, we thought if you could make messenger RNA a drug, what you could do is you need to protect it. That's where you need the nanoparticle. But if you inject it in the body, the body's the factory. The body will make the protein. So you could do this very, very quickly. In fact, we started this company in 2010. By the end of 2019, we had 13 different products in human clinical trials, and then COVID came. Next slide. And before I do that, I just want to mention there's a lot of other diseases you could treat too, like intracellular or membrane-bound proteins you could make this way. At any rate, like I say, we worked on this at Moderna for about 9 years, and then COVID came in late 2019, early 2020. And let me just show you how fast Moderna was able to make the vaccine. It was pretty much unprecedented. Next slide. So the Chinese scientists published the virus genetic sequence on January 11th. It took less than 2 days to finalize the design, put it in the nanoparticles, and within a month, we had shipped batches to our clinical partner, NIH, for testing. The first patient was treated in what's called a phase 1 trial, literally about 63 days after we started, 64 days. And by May 19th, we had the data on the phase 1 trial. Just so you know, in case people aren't aware, when you do a clinical trial, usually you do escalating dose to find what the highest safe dose is. So what we did is we had that data by May 19th, and we could see what dose was safe, but also you can get significant antibody titers, and you could find out if the vaccine was likely to work. So, when Moderna announced that data, I thought that was pretty exciting. Nonetheless, the news media and many scientists were, just like they were always critical of me when I started, they were very critical of Moderna. The next slide shows the Boston Globe, my local newspaper, their headlines on May 19th. And their headline was, "This is not how you do science." And you can see whose picture is on the cover, front page, me, which was unfortunate, but that's the news media. At any rate, so I want to go just 6 months after that. So after you do a phase 1 trial, then you do a phase 2 trial, and finally you do a phase 3 trial. So we started that. And by November, That trial, which had 30,000 patients, was done double blind, we broke the code. And I remember being in the boardroom. So 15,000 get placebo, 15,000 get our vaccine. And it was remarkable when those data were announced. Not a single person of the 15,000 died. Not a single person of the 15,000 even went to a hospital. And it was 94 to 95% effective. And of course, next slide, it's now been used all over the world. Another one in nanoparticles by BioNTech and Pfizer also has been used all over the world, probably several billion people. Let me just show you quickly some of the data. Next slide. So this is, showing you what happens between unvaccinated on the top, vaccinated with no booster, and then vaccinated with a booster. Let me show you on the next slide the effectiveness of the different vaccines. The Moderna one, 94%, Pfizer and BioNTech, 91%, Johnson & Johnson, 66%. And then that got pulled from the market. There's multiple articles published in the past year about the efficacy of these vaccines. I'll just show you two very large studies in the New England Journal of Medicine. On the next slide, you can see almost 400,000 patients that if you look at the incidence of COVID infection, it's by far the lowest in mRNA 1273, very, very tiny percentage. And if you go to next slide, you're going to be looking at hospitalizations. And again, it's like even after 168 days, like 0.05% for mRNA 1273. It's a little bit higher for the BioNTech one, but that did very well also. Not only can you use it for these vaccines, and they continue to be updated with boosters, but recently Moderna announced, next slide, that you could do what's called a personalized cancer vaccine. That we call that PCV. And the idea, this is with late stage melanoma, 157 patients, you could take what are called neoepitopes, and you could take the top 34, and you could design mRNAs that will make proteins and antibodies and immune cells that will attack them. And what happened in this case is, in this trial, half of them got Keytruda, which is an immunotherapy drug, half got Keytruda and the personalized cancer vaccines. And the results were pretty amazing, 44% reduction of recurrence or death. Now, the last thing I was going to talk about is I was visited way before I did this by a very successful software entrepreneur. You see me on the left and him on the right. If you're not familiar with who that is, it's Bill Gates. And Bill has always been very interested in vaccines. And we had worked on vaccines with the idea of nano and microparticles now since the 70s. But one of the things that he and his team wondered about is, and he was concerned about people in Africa, that they didn't come back for injections. So if we could go to the next slide, I'd want to show you what he asked. Next slide. Could we come up with a single injection, but get pulses, like at time 0, 1 month, 2 months? So we'd like make a cocktail. If you look in the middle of the bottom, where you see that red box covered with gray, it says micromolded particles. Our idea is that by changing the composition of the gray part by different degradable polymers, and they'll degrade into water and CO2, it's polylactic glycolic acid, we could make them burst at different times. So these could be like little tiny eggshells, but inside the shells, they would have the vaccine, and the shells would break. And when they break at whatever predetermined time we want, you'll get those bursts. So that's what we wanted to do. And we even developed a whole new printing process. And I'm going to show you another video of how we did that. Next slide.
The stamped assembly of polymer layers, or seal method, combines computer chip manufacturing technology with a novel layer-by-layer assembling method to create an array of core shell polymer particles. First, the polymer of choice, PLGA in this example, is melt-pressed using a prefabricated silicone mold. The mold with the particles is then transferred to another substrate, where it is peeled off, leaving behind an array of polymer-based particles. These are then filled with any drug or vaccine using an inkjet piezoelectric nozzle and then dried. To seal the filled particles, a cap mold is aligned, sealed with the base, and delaminated. The resulting array of core shell particles are then removed from the base and stored until use.
So we made these. We actually published this in Science, and we published 5 follow-ups in Science, other science journals on using these for cancer and improving them and so forth. We also filed a patent on this, and let me just show you some of the data on the next slide, which is very exciting. This is going to be just one injection. If we could go to the next slide, and what you see is 11 bursts, all at different times, from like 1 week to 6 months. And you can do that by just changing the composition of polylactic glycolic acid and its molecular weights and stuff like that, because that will make the shells break at different times. And on the very final slide, what we're doing is not only testing this by injection, but also, if we could go to the last slide, we're working with Mark Prousnitz, another one of my students who's a professor at Georgia Tech. And what we're doing is we're putting them into microneedle patches. You see a close-up of those microneedle patches on the right. But the beauty of these patches is the needles don't go in very deep, but they go in deep enough so that they can do the injection, but not so deep that they hurt. So they actually look like a little Band-Aid if you go to the left and on the screen. And then in the middle, you see somebody just putting it on the skin. And our goal, is to use these for vaccines someday all over the world. That's what the Gates Foundation wants. That's what we want. And so that, I hope, will end up helping even billions more people someday. And there's all kinds of vaccines, not just mRNA vaccines, but we've been working on polio vaccine with the Gates Foundation and many others as well. Thank you so much for having me. I am happy to answer any questions.
We now invite Prof Misery to moderate the Q&A session.
Okay. All right, can you hear me? Okay, well, thank you, Bob. This is Ali Misery from NTU. Thank you for this great talk. We never met directly, but we just got a paper together last week. So I can be part of your network now. I would like to start with the first question, if you don't mind.
Of course.
You have mentioned in your talk and your paper that people would not believe in your approach and proposal. So I would like to return the question to you. Have you ever come across some technology that you're not convinced at first, and then it proved you wrong eventually?
Yeah, well, I'm sure I have, but the way I look at it, because of that experience, I have so many people coming to me with ideas, mostly my own students, but and my feeling is to never tell people you can't do something. I try to say, well, it might take a while, or or maybe you should think about doing this or that. But I don't think I've ever really, because it was so damaging to me, so discouraging to me to have all these people tell me it was wrong and turn our grants down. It really had a big effect on I think how I think about things today. That really, I don't think anything is impossible. You just don't know how long it may take. So but I'm sure there are things I probably felt might take a lot longer than they did.
Okay, well, thank you for this positive message. I'm sure many PI will take it in a very good way. Maybe I will go with some of the online questions here. So we have one here. Which one do you see here? Well, I'm not sure, but here we go. I think this is this one here. So do you think your chemical engineering background and knowledge contributed to your achievement in nanoparticle-based drug delivery?
Oh, absolutely. I think chemical engineering really was a great background for that. I mean, I learned about materials, I learned about polymers and lipids. And I also think chemical engineering, again, maybe I'm prejudiced, but I think it teaches you how to think in a very constructive way about problem solving. So I think both the specifics and the general things that I learned were very helpful. And I also think that it's funny, when I first started doing my PhD thesis, I remember there was a really good postdoc there, Colin Gardner, and he just really drilled into me about doing things really reproducibly. He had me do standard curves with like 30 data points over and over again, so I'd get almost like 1% error. And so I think that the whole experience of doing a chemical engineering doctorate was very, very helpful to me. I have to admit, going to a surgery lab after that was like very, very different. But I think I do think the question is a good one, that it probably helped me a lot.
I think we have a question from the audience up there.
Morning. Thank you, Prof. Roberts. My name is Lorato from the University of Cape Town in South Africa. I'd firstly really like to thank you so much for your talk this morning. I feel extremely encouraged and inspired by your journey. I'm particularly really curious about what kept you going with the extreme level of rejection that you encountered from such a young, from your young days in your journey. What really went through your mind every time you were told, especially when you were trying to prove the concept of nanoparticles and angiogenesis, anti-angiogenetic agents? What kept you going and believing in the idea that you had? And what kept you, trying out the experiments, irrespective, despite all the, grant application rejections?
Yeah, well, that's a very good question. I think there are a couple things that were probably helpful to me. One is I think I was very lucky that my postdoctoral mentor, Judah Folkman, he kind of went through a lot of the same things. People didn't believe in what he was doing, and and yet he kept trying and he kept believing. And I think having role models is a really good thing, and he was a great role model for me. I think separate from that, maybe it's like my upbringing, my parents, I think, I grew up my dad, ran a tiny liquor store in Albany, and he worked all the time. And I think that was, a good also role model for me to see. And then maybe some of it's just genetics. I'm just a really stubborn person, I think, so but I think it's maybe the combination of all those things. But I think, like I say, for so many people, including me, having role models is very, very helpful and important.
Thank you. And just the last question, regards to the mRNA vaccine, great work, made a tremendous impact on how the pandemic eventually got regulated. What are your thoughts around access and how everything played out regarding the COVID vaccine access, particularly in African countries? Because as we know right now, I think only 30% of Africans are actually vaccinated, and there's been a lot of politics around access and patencies. What are your thoughts around that? Because for me, I found it extremely discouraging, because we have these meetings where we see great inventions and scientific innovations, but at the end of the day, when it's implementation, only certain regions in the world benefit from this great, exciting scientific innovations. What are your thoughts around that?
Yeah, well, I think that's a great question. Let me make a couple points on that. And again, you may know better than I on some of these. But, like I say, from our MIT lab, one of our big efforts, even way before this, has been exactly what you're saying. In other words, to try to, make vaccines, accessible to everybody and to ship them around the world. But you're right, when this crisis came, then it was difficult. And I don't know all of the different things. In other words, one of the things, like, for example, Moderna, it depends at what point in time. Moderna actually tried to give, I think, over 100 million doses away, and they got turned back. And my understanding, and again, like I say, you may know better than me, and other people may too, but a lot of it wasn't just the access, wasn't just getting the vaccines, it was also the journey, whether it's from the air, people would say the journey to people's arms. In other words, some of it had to do with the logistics, I guess. Even if you could get them to Africa, apparently, like I say, when Moderna tried to give them to some of the African nations, they could get to an airport, but then somehow they wouldn't get to the patient's arms. And whether it's the roads or other logistics, I don't really know. But my sense is that there needs to be more work done across the board, not only the politics and ways that you could ship them, but and again, like I say, you'd know better than me whether there's things to do with the roads or customs or other things. Like I say, you may know more than I do.
Thank you. I think what Robert is alluding to is also the issue of cold chain management, which, as far as I know, takes 80% of the cost of a vaccine. And that's sometimes the issue of going to the right location, remaining in the cold chain. I think we have some question up here.
Hello, sir. I'm from the Hebrew University of Jerusalem. Thank you for your talk. It was an absolute privilege to hear from you. And my question is a bit more technical also about the mRNA vaccine. So you're talking a lot about the technique of how the cell will be the factory and how the mRNA will be delivered, but how can we control the 3D fold and the epitope that is being presented to the immune system and prevent phenomena such as antibody-dependent enhancement of infection?
Yeah, well, that's certainly a very good question. I mean, I don't know that I'm necessarily the best person to answer that. You know, it seems to me that the vaccines have worked quite well, but how you control all these different things, I mean, it's so much of it has to do with the body's own machinery, which is what we're relying on. And of course, not everyone is the same. But I don't really have a great answer for looking at the protein folding after you get the mRNA into the cell. I wish I knew more about that.
Thank you.
We have another question from the audience.
Yes, hello. That was very fascinating. Thank you very much. Very inspiring talk. I also have a rather technical question. I mean, we just talked about, let's say, the delivery issue of the vaccines, for example, and I was very intrigued by the very last part where you show these kind of single-shot injections with slow and fast-releasing particles. So I was wondering, I mean, I'm really not an expert. What happens, let's say, to the slowly releasing ones inside the body? I guess they are also somewhat peg-coated to prevent clearing, but where did they spend these, let's say, 1 or 2 months? And is there some kind of other clearing mechanisms that could make it complicated to be applied?
I hope so. I think the answer is I hope it won't be that complicated. We're really relying on things that we and others have done before. So the idea, just a couple of things, you know, and I showed the earlier some of that picture with all the different drugs. A lot of them were made out of what's called polylactic glycolic acid, and they would deliver in that case like anti-schizophrenic drugs, anti-cancer drugs. And what you do when you do those is you inject them underneath the skin, like intramuscular, you know, with a needle, underneath the skin, and subcutaneously or intramuscularly. And in those cases, the system just stays there, but and drug diffuses out, and the polymer in that case, polylactic glycolic acid, dissolves. But it just stays there. It just stays there. It dissolves. The drug keeps coming out. And in essence, that's kind of what we're doing here. Basically, we'd have different polylactic glycolic acids. They'd all reside in that subcutaneous or intramuscular place, but at different time points, they would dissolve completely. You know, like I say, we could make it dissolve in a week, but we could also make it dissolve in 6 months. So we're giving this cocktail, but they'd all reside at in that place in the body where you'd inject them, like you get that injection right now, probably in your arm, subcutaneously or intramuscularly. And so they would just reside there. By the way, also, just so you know, the beauty of that polymer, lactic glycolic acid, it ultimately will degrade to water and carbon dioxide. So everything goes away.
I think we have time for one quick question because there is 40 seconds left. I have one online question that I can quickly ask you, Bob. Someone is asking, do you think mRNA vaccine can be used to treat neurodegenerative diseases? What are your views on this?
Yeah, so let me give you 2 parts to that answer. I've had that question before. I mean, the question is absolutely, but I remember I was giving a talk to Glaxo's board, and somebody asked me that very question: how can we treat COVID but we can't treat Alzheimer's? And the answer to me is we don't know the biology well enough, right? We know that the reason we can treat COVID in a way and have a vaccine is that we understand the biology. You go after the spike protein. Unfortunately, in the case of Alzheimer's, the biology is not nearly as well understood. There are other problems too, like delivery across the blood-brain barrier, but not understanding the biology when you don't understand it, I don't know that mRNA or anything's going to be that helpful. You need to understand the biology first, in my opinion. So says somebody who's not a biologist.
Okay, well, on this note of supporting basic science, thank you again, Bob, for your wonderful talk and answering all those questions, and thank you everyone for your attention. Thank you.
Thank you.
Thank you, Prof Langer and Prof Misere. Similar to yesterday, we have an exciting lineup of online engagements with this morning's session by Singapore's Centre for Quantum Technologies on the topic Quantum Computing Atom by Atom. Do log in to the GAVME platform to attend the online talk. In the meantime, we invite you for a short break with refreshments outside. Kindly be back here for the next plenary at 11 a.m. See you all in a bit.
Good morning, everybody, and welcome to the first online engagement session of today's program. My name is Jenny Hogan. I'm the Outreach Manager for the Centre for Quantum Technologies here in Singapore, and we'd like to thank NRF for inviting us to participate here with you today at the Global Young Scientist Summit. So we're very lucky to have with us this morning Prof Lo Huan-Chen, who's going to talk to you in just a moment about quantum computing. First, I'm going to give you a little context on the Centre for Quantum Technologies, which we call CQT for short. So CQT was established back in 2007 as Singapore's first research centre of excellence. We're now 15 years old. We have some 230 staff and students who do research across 3 application areas of quantum technologies. So that's computing and simulation, communications and sensing and metrology, as well as developing advanced instruments and continuing with curiosity-driven basic science. We're hosted at the National University of Singapore, but we have researchers too at the Nanyang Technological University and with the Agency for Science, Technology and Research. So many of you in the audience may know about quantum technologies. It's a very fast-growing field around the world. And here in 2022 in Singapore, they've established a national quantum office that will be guiding the country's national strategy, and CQT will be supporting this effort to grow the research and industry ecosystem here. So we already have a few quantum startups even here in Singapore that are founded by our alumni. But I think we should get to the science. So our research is led by 25 principal investigators, and Prof Lo is one of them. Huan-Chen's also a President's Assistant Professor appointed in the NUS Department of Physics. And for her research, she's been recognized as an NRF Fellow in the class of 2018. And in 2020, she was named as one of 15 of L'Oreal UNESCO's International Rising Talents worldwide. She's also serving on the World Economic Forum Global Future Council for Quantum Computing. So she'd started her research career as a PhD student in the US in the lab of Nobel laureate Eric Cornell, and now she's leading her own group here in Singapore. So those of you participating in GYASS, you can ask questions during the talk by typing them into Slido, and we'll be keeping an eye on at the end. So Huan-Chen, I'd like to pass over to you.
Okay, thanks Jenny for the introduction, and thanks to NRF for the invitation to give a short talk here. Today I'm very excited to share with everyone some of these basic ideas on quantum computing. And then I plan to run through one of the leading quantum computing platforms, which is with these arrays of neutral atoms. That is also the platform that I work with in my laboratory here at the Centre for Quantum Technologies in the National University of Singapore. So this is supposed to be a fairly short talk, and I look forward to having lots of questions from everyone. So let's go to the very basics. In classical computing, we encode data using bits of zeros and ones. Now, in quantum computing, we also use strings of bits, but each of these bits can behave like a quantum bit, or what we call a qubit. Right? So a qubit can be in state 0, or it can be in state 1, or it can be in both at the same time. So now, quantum mechanics describes basically the world of the very small, or the very cold. So suppose, let's imagine you have atoms as your qubits. So you may have noticed that here I indicated the state 0 as this atom being in the lower state of 2 states. Or this atom can be described as behaving like a little magnet that points upwards. In any case, either of these pictures works just fine. And the idea, once again, is that this qubit can be in both states at once, or what we call a superposition of both states. On the other hand, the power of quantum computing doesn't come from having just a single atom being in a superposition state. Rather, this power comes from having many atoms that can be put in some collectively entangled state. Now, this entanglement is a funny thing. If you have 2 particles that are highly entangled with each other, it means that the moment you measure the state of one particle, you immediately know the state of the other particle with a much higher degree of correlation than you can ever explain classically. So what is this quantum computing good for? Well, some of the famous examples include trying to factorize large numbers, which is of course important for breaking today's RSA keys, or you can have a more efficient search algorithm. And the governments recognize the power of having such quantum computers, which is why today there is really extensive effort in many countries around the world to pursue quantum computing research as a strategic trust. However, quantum computers that can perform such tasks are not yet on the horizon, because in order to do so, you require millions of qubits that can perform these gate operations pristinely, which means at a much lower error rate than what can currently be achieved. In fact, one would say that today we are living in the NISQ era, which means that quantum computers today are these noisy intermediate scale quantum devices. Okay, so they are noisy and they are not that large yet, but somehow even when we have these NISQ devices, there has been tremendous interest even from industry to jump onto quantum computing research. So here's a list of some of the companies that are involved in quantum computing. Of course, you've got the big tech companies, to those that are in the transport, energy, and finance sector, to startups that pursue quantum computing, including, of course, some of our very own spin-offs here in Singapore of CQT. So there must be interesting problems that can be solved by these NISQ devices, and that is what I want to focus for the rest of this talk. So to appreciate the kinds of problems that are tackled by these NISQ devices, let's take a different approach. So we'll take a look instead at the problems that take up the most computing resources of these classical supercomputers today. Now here's a pie chart of one of these supercomputing centers, for example. And many of those problems, as you can see here, are materials research and chemistry problems with some engineering and various optimization problems involved here. So what happens with these classical simulations, right, is that you have very clever theorists who come along and write down a model of the problem that is to be solved, input those equations into a supercomputer, which then spits out the results after some time. However, some of these challenges require solving quantum mechanical problems, which are notoriously hard to calculate. For instance, if you want to exactly solve for the time-dependent behavior of a collection of even a few hundred atoms that are strongly entangled with each other. That can be a very challenging problem because the number of classical bits that are required to completely, exactly model this system would be more than the number of particles you have in the whole universe. Okay, so that's a problem. And the solution to this problem can be found in these quantum simulators, which are what many of these NISQ devices can act as. So there's a famous physicist, a Nobel laureate himself, Richard Feynman, who once said that nature isn't classical, right? So if you want to make a simulation of nature, you had better make it quantum mechanical. By golly, that's a wonderful problem because it doesn't look so easy. So the idea here is to use this simple quantum nanomachines, right, the NISQ devices. And these are physically like atoms, molecules, photons, which are particles of light. You use them to simulate more complex quantum phenomena. And Feynman is right that it's not so easy, right? Because these simulators need to be controlled, isolated, and yet tunable. However, once you can set up these simulators to obey the same equations, right, as those that model the complex phenomena, then you can simply perform your analog simulation by letting your atoms or molecules evolve in time. And then you can read out the answer by making measurements on the states of your atoms or molecules. Now, to see how quantum simulators work, let's consider materials as an example. Right? So in general, the macroscopic properties of materials map onto the microscopic behavior. So for instance, electrical conductivity is simply related to how charges, so electrons, move in time, and thermal conductivity is related to how phonons move in time. So if I'm allowed to now think of materials as a collection of atoms, right, they are arranged in a particular underlying geometry, and if I can induce tunable interactions between them, then I can dream of building up these analog materials with my quantum simulators as these Lego blocks. And these are built up block by block. And then the dynamics of these Lego blocks are then related to the macroscopic phenomena that I mentioned earlier on. So here is actually a list of some of the very promising candidate platforms for quantum simulation and eventually for quantum computing. So you've got trapped ions, superconducting circuits as two of the big candidates. And of course, you've got optical lattices, quantum gases, photons, and so on. Now, each of these platforms demand being able to control the individual quantum bits in a scalable manner, because remember, we need to eventually scale this up to hundreds, thousands, millions. And you need to program them in some way. So there must be some inherent degree of programmability of these platforms. And today I'll focus here actually on the atom arrays. And there are many nice degrees of freedom that one can control with the atom arrays. So here, for instance, you can control the internal quantum states, the external motional states, You can arrange these arrays in different dimensions, right? 1 dimension, 2 dimensions, even 3 dimensions. And then they can be arranged in arbitrary geometries with user-defined doping, something that is not very easy to do in an actual solid-state system. And finally, you can also use lasers to induce tunable interactions over long range between these atoms. So in the last few years, atom arrays has actually really taken off as a platform of choice in the quantum physics community. And one of these key enabling techniques that allowed the atom array platform to become so popular was really this ability to control individual atoms in their optical traps by mapping the optical domain onto the radio frequency domain. So these are arrays of atoms that are trapped in individual tweezers, and the tweezers are formed from tightly focused laser beam. Now, if I want to have 100 tweezers, does that mean that then I have to build 100 different lasers? No. There is this magical device over here, for instance, that you can use, an acousto-optic deflector. And what that does is that it takes an incoming laser beam and it sends in a radio frequency wave, and it sort of kicks that laser beam off to one side at a given angle. So if you've got a given deflection, now that means that if you send in 100 simultaneous radio frequency waves, you can actually have 100 simultaneous deflections, which then get mapped onto the atom plane as 100 tweezers. So this is nice because all you have to do is to start off with a laser beam that's 100 times more powerful, which is much easier to achieve. So here you see an example actually of atoms, single atoms, in 400 traps, right, formed from 20 by 20 array. And that is the average fluorescence that you get. However, one problem is that if you have 100 traps, 400 traps, it doesn't guarantee you 400 atoms, because it turns out that the loading of single atoms into a tweezer is probabilistic. In fact, due to an understanding of atomic physics, that probability is about 50% or so. So the probability of loading a single atom into a tight trap is the same as getting a hit when you flip a fair coin. But now the problem comes in the following. When you perform your quantum simulation or your quantum computation, you have in mind a pristine atom array to begin with. So it should be defect-free, and you want to start off with 100 atoms. That means there should be no holes in between. However, as you can see, where you want to have 100 consecutive hits when you are given 100 coins, the probability of achieving that is really small. It's 0.5 to the power of 100, which is 10 to the minus 30. That's just abysmal. So we need a solution around this. And this can be done simply by starting with 200 coins, right? Flipping them and getting 200 hits, and then very quickly shuffling those hits together such that you have your subset defect-free array. So with this shuffling around of the atoms, okay, this can allow us to get a substantially higher probability of having defect-free arrays as a starting point for these quantum simulations. This can be done by simply overlapping a second set of mobile tweezers that pick up the atoms in real time and shuffle them. And real time basically means that this happens faster than a blink of an eye. So here you see a GIF image of actually 2 consecutive sequences, images that are taken of the atoms. So the first one is the initial loading, and you can see that there are many holes in between with the defects. And the second one, after tens of milliseconds, is your defect-free 225 atom array. And in fact, this defect-free array that you're looking at is pretty much one of the largest that you can get right now in the world. So going ahead, we've also demonstrated actually in our laboratory that we can apply this algorithm, rearrangement algorithm to various different arbitrary geometries. For instance, you've got the honeycomb, Kagome, link Kagome. These are all interesting for studying different advanced materials like graphene, and for studying superconductors or quantum spin liquids, like this Hilbert-Smithite crystal over here. We've also demonstrated that we can use this to potentially study fractals that are basically quite interesting as fractals occur in nature. And also just to show that we've done this in Singapore, we also rearranged this to form the lion head symbol with the single atoms. So as an outlook, here we are marching towards trying to understand advanced materials. So you can really now develop a microscopic detailed understanding of what is going on when you set up your spins to model a particular phenomena. Or actually now what is very hot in the atom array community is to use these atom arrays to tackle optimization problems such as these new classes of NP-hard problems like the maximum independent sets, which actually have direct applications in the placement of radio antennas for optimal network coverage, and so on. So with this, I kind of want to close by saying that, so far I've described using quantum physics to impact quantum computing and simulation. But there's really a range of quantum technologies, as Jenny mentioned in her introduction. There can be more precise quantum sensors that can be used based on quantum physics. And you can also use these for quantum communication, where the security is really guaranteed by the laws of quantum mechanics. So yeah, with this, I think I end my talk here, and I'm happy to take questions.
Thank you, Han-Chen. So just to remind everyone who's joining from the Global Young Scientist Summit, you can send questions in through the event platforms and type them into Slido. So we'll be looking there and we'll put your questions. So Han-Chen, maybe can you tell us a little bit about what's the next challenge for your research?
Yeah, so so far we have gone to realize basically one of the largest atom arrays. You can of course try to scale them up even further. Now that's not a straightforward task. There are sort of new architectures that might have to be developed, still involving lasers and atoms. But another direction now is to really try to induce interactions between the atoms that you have now generated, because these interactions are really the key to having some programmable features to be able to map them onto the desired equation that you want to solve, and try to understand models of quantum magnetism or other interesting aspects of materials. Yeah, I should also mention actually, in case anyone's interested, we also have a virtual lab tour to offer for our lab, so you can see what actually these experiments look like in real life.
Thanks, Wen-Chen. So although it looks rather simple when you show a picture of an array of atoms, I know from visiting your lab that you have a few large tables full of equipment, lasers and lenses. So you can look for the virtual tour of the National University of Singapore College of Humanities and Sciences to see a sort of 360-degree panorama of all the equipment that Wen-Chen's group has built up for these amazing experiments controlling single particles. I wonder, Wen-Chen, so you came to CQT in 2017, and that was when you were starting up your group with the NRF Fellowship. For young scientists in the audience, could you maybe tell a little bit about what was exciting and maybe what was challenging to make this transition to running your own lab?
Yeah, so I think it was really very exhilarating to be able to pursue my own research ideas. So these are ideas that you've sort of formulated over the years, and also they evolve as they go along. And it's nice that you're now able to chart every single thing out, right from deciding in your laboratory how it should be renovated, where the optical tables should be placed, to finally deciding what the atoms should be doing, what's the best transitions to address them, things like that. So I found all of that really exhilarating. And of course, it was really nice to be able to have the NRF Fellowship to back all of this up, to really transform the ideas that we had into actually, the physical setup. And it was also very exciting to be able to build a team. Sometimes I feel that, starting off as your own laboratory, it almost feels a little bit like starting a company, right? You have to find your own, you have to find funding, and you have to be able to talk to students and grow the people in your team. So all that was really very exciting, because every day there were just new things that had to be discovered in all sorts of aspects.
And you have how big is your group now? You have quite a few people working in this from different backgrounds.
Right, so we have 12 people in my group right now, split across 2 experiments. We're almost a little bit constrained by lab space, but the background that they have, it's quite diverse. There are people who, there's firstly in terms of educational background, there are people who already come with PhDs, postdocs, all the way to undergrads, whom we are very happy to take on and mentor. Many of them are physicists, so they come from a physics background. Not all of them start off with laboratory experience. Some of them even might be more interested in doing theory at the beginning, but then they want to try experiments. And also we've taken on engineers as well, who really come in with a very different perspective on how things could be built better. And building things robustly is really a part of our culture, because you need these atoms to be scalable and so on, and that does not work if things are always falling apart. So it was really nice to be able to tap on many different talents in the group.
Okay, well, I'm pleased to say that we have some very good science questions coming for you on Slido here. So there's maybe two that can be taken together. So one question is someone who says they're from biology. So how do you use these systems to study materials if the material aggregated into single atoms? And perhaps related, you'll be able to answer a question that's come from Edelman Jan in Switzerland, that how do you study the interactions between the atoms in the different tweezers?
Okay, so those are all great questions. So how we make the material is we really make a model of the material. So we're not claiming that the collection of single atoms that you work with, maybe if I pull up over here, an example over here, for instance, of the honeycomb, right? We're not saying that these honeycomb of atoms that we trap are exactly used for graphene. However, that's where physics comes in. Physics sort of distills the phenomena that is happening behind that has been observed with graphene, and we try to come up with models for those. And usually those models are the ones that then get input into computers to really solve exactly what are the dynamics of what is going on, and try to see whether that maps and that matches the experiment. But it turns out that some of these models, to be able to be solved exactly, are just very difficult, and it's too challenging for classical computers to take on. You can think of it as saying that there are too many parameters for the simulation to be able to handle. So this is where quantum simulation comes in. So we're not claiming that this is then going to be used as your graphene nanotube material that you take along with you in your pocket, but really this is allowing researchers to be able to develop more accurate models, such that now we can say, okay, with these particular properties, then that comes from these parts of the equation. Now, if you tune it in this way, what new things can we expect? And then we use those to hopefully guide the design of new materials that way. Yeah. So related to that, in terms of the interactions, so these are tweezer-trapped atoms, and we actually induce interactions between them by driving them to the Rydberg states. So Rydberg states are basically highly excited atoms. So you can think of sort of a very floppy atom, if you like, and that they can have a huge electric dipole moment, which then allows them to interact over large distances with a fairly high frequency. So just to put on some numbers, it's something like 100 megahertz kind of interaction strength, which is considered huge over a few microns. So with these Rydberg interactions, they're tunable. You can change the range of the interactions. If you drive two of them to Rydberg states, you can even have dipolar interactions between them. So in other words, they can be attractive or repulsive. Well, repulsive or attractive, depending on how they align relative to each other. And there have been many interesting things that have been discovered, and many more with these kinds of Rydberg atoms.
Okay, well, I hope this is answering the questions for the audience. We probably have time for one quick answer to one last short question. So V. Vijendran from Singapore says, when you scale up the optical setup for larger atoms array, does the computational cost of optimizing the experimental setup grow prohibitively? Maybe why, what's the limit on the size that you're dealing with?
Right, great question. So the computational limits of the possibility of using these arrays would basically scale exponentially. So it's rather the computational promise, is what I want to say. So if you really could realize, 1,000 atoms instead of 100 atoms, well, that would basically be, e to the 10 times more higher computational power that you would get. So that is very promising for tackling those very complex problems and so on. But in terms of building the setup, the beautiful thing is that you do not need that much more to be able to scale up, precisely because of some of these devices that I've talked about previously, where you're really mapping the optical problem into a radio frequency problem. And so because the frequency has come down by so much, it is much easier to control. And in a way, to some sometimes you might be limited by the bandwidth, but you can always also stack them in different dimensions, which also allows you to scale up quite significantly. Yeah.
Okay, thank you. I'm afraid we're going to have to end our session here with so that we can return the GYSS participants to the live audience. For those, if we didn't get to your question, or if you had questions you didn't get a chance to ask today, I mean, please look us up online. So CQT is Quantum La on the web and on social media, and Huan Chen, you'll be able to find a group page there too. So yeah, thank you, Huan Chen, for the talk. Thanks, NRF, for inviting us, and thank you to the audience for listening.
Yeah, thanks to everyone.
Welcome back, everyone. This morning's program continues with Prof Hartmut Michel, who will talk about how structures of intermediates of the cytochrome C oxidase reaction cycle suggest a revolution. The Q&A session will be moderated by Prof Oliver Mulakaya. Prof Michel, please.
So, thank you very much. And it's really a pleasure for me to be back here in Singapore. And actually, I work at the Max Planck Institute of Biophysics in Frankfurt am Main, Germany. There are about 200 people, and we all work on membrane proteins because we think that the membrane proteins are the most important subject to study. And I show you here a picture of eukaryotic cell, and everything you see is membranes. So you have a cytoplasmic membrane, which surrounds the cell. You have membranes for the organelles, for the mitochondria, for instance. We have the lysosomes, the waste recycling stations. We have the Golgi apparatus modifying proteins. And we have the nuclear membrane, where you have the DNA, the library. And what is the problem? That the membranes are barriers, and you have to transport substances across the membrane. And including ions, you have active transport, you have passive transport. And also what is very important in membranes, that biological electron transfer in photosynthesis and cellular respiration occurs in biological membranes. And this is my major focus of my work. You have already heard about signal receptors. Yesterday, a beautiful talk by Brian Kubilka. And signal receptors like GPCRs, they transduce the signal, amplify it. And what is very important is that 80% of all marketed drugs act on membrane proteins by either inhibiting them or by activating them. Some of membrane proteins are enzymes, preferentially if the substrate of the enzyme is hydrophobic. And we want to get the atomic structures. This means we want to have the position of each atom, and we want to see the changes of the atom position when the membrane protein performs its action. So this is what we call understanding the mechanism of action. And you need that information also for rational drug design or virtual screening, as also was outlined by Brian Kubilka yesterday. And so my interest is how to get atomic structures of these proteins. And nowadays we have 5 methods to do that. So we have X-ray crystallography, mainly due to the work by Max Perutz already in the 60s of the last century. And this is mainly what I also used. Next method which came was nuclear magnetic resonance spectroscopy, which works for proteins up to 30,000. We have electron crystallography. There are only 2 or 3 examples that electron crystallography could be used to determine structures of proteins. We have neutron crystallography. I would like very much, but you need a neutron source, very expensive, and you also need huge crystals, maybe 1 centimeter in each direction, because in order to get really diffraction of the neutrons. And nowadays, the methods of choice is single particle electron cryomicroscopy. But when I started in the 70s, the only method was X-ray crystallography. And this caused a problem because the opinion was it is impossible to crystallize membrane proteins. So due to that opinion, people didn't start the project. Nevertheless, I tried to do that, and I developed some ideas how one should be able to crystallize membrane proteins. For that, I show you here a basic picture of a membrane, biological bilayer membrane. You have the bilayer of the lipids with the polar head groups of the lipids on both sides of the membranes, and you have the fatty acid side chains here. And you have the membrane proteins incorporated, and the problem is that they have a hydrophobic surface where they are in contact with the alkyl chains, and they are polar where they are in contact with the aqueous phases on both sides of the membrane. And what you have to do is you add detergents, soaps, forming micelles. The micelles take up the lipids, and also they take up the proteins and form something like a safety belt around the hydrophobic surface of the protein. And that's a situation where you start to purify and to crystallize a membrane protein. And I came up with 2 solutions for how to crystallize proteins. One I call type 1, that you get 2-dimensional crystals in the plane of the membrane, and you can form stacks of these membranes, and you have something, a very nice crystal, but this was out of reach at that time. I focused on the type 2, where you try to crystallize the membrane protein within the detergent micelle, and the major problem is that you have to optimize the detergent micelle in size and shape and chemical behavior. You also have to have some attractive interaction between detergent micelles in order to be able to get a well-ordered crystal lattice. And then you need, of course, a protein, and I went to photosynthesis. This primarily because these proteins are nicely colored, so it's really a joy to work with these colored proteins. And color changes indicate whether the protein is happy or unhappy. And also they are stable and they are available in huge amounts. And I took a photosynthetic purple bacterium called Rhodopseudomonas viridis, and full of photosynthetic membranes consisting of light harvesting complex and photosynthetic reaction centers. Isolated them, and after about 3 years, I got crystals, and the crystals, one of the crystals is shown here. And what was very nice from the beginning of it, that we could get very nice diffraction patterns, and so that's a rotation photograph taken at a rotating anode source at home. And this photograph films to record the data, and you need about 120 photographs like this, each taking about 12 to 14 hours, 10 to 14 hours. And so it took me to collect one data set 3 to 4 months. And you need several data sets in order to find also the heavy atom derivatives. And nowadays we have the revolution, we have synchrotron radiation, we have cryocooling, we have modern detecting devices. And this means now when you go to a synchrotron with a tiny crystal, you get the same data which I used to collect in 4 months within 8 seconds. Can you imagine? Change from 3 to 4 months of work to 8 seconds. So this is really revolution and make the make them much faster, and you need much smaller crystals. And nevertheless, we could determine the crystal structure, and you see here the photosynthetic reaction center from that bacterium, and this is incorporated into the membrane. You have two protein subunits. Surprise was the whole structure is highly symmetric, and so the two protein subunits here are derived by a gene duplication from a symmetric reaction center, very clear. You have here scaffolding protein, and here protein cytochrome C, which delivers electrons back to the reaction center. If we look to the photosynthetic pigments, we have here the primary electron donor, which is also called special pair. When it gets excited, an electron leaves the donor, jumps onto bacterial chlorophyll, then onto bacterial phytin, then on the first quinone, and then on the second quinone. And this branch is not used for electron transfer, maybe used for light harvesting complexes, but it's still there. Evolution did not get rid of it. And we find this theme in all photosynthetic reaction center cores. Now people were able to crystallize and get structures of all different types of photosynthetic reaction centers. And so this is the purple reaction center, where they have the primary electron donor, the one branch which is used the other way around. So this is the two protein subunits, the L and the M subunit. This is photosystem 1, where you have also this kind of primary electron donor, then the chlorophylls and the quinones at the end, and then here you have iron sulfur proteins to go on. And so the cores of the reaction centers are all very similar, and this means that photosynthesis was invented only once during evolution. So I wonder whether this also would happen on a different planet quite far out of the universe, or how unique this invention of this photosynthetic reaction center was. So, and now with respect to applications, you always asked, can we use photosynthesis in order to solve the energy problem of mankind and to reduce global warming? Because in biomass production, you take out carbon dioxide out of the atmosphere and should reduce the global warming. And so some basic facts: we have the light reactions called by the light absorbing antenna pigments, and the transfer of the energy to the reaction center. And in photosystem 2, you get oxygen, you get water splitting and oxygen release as waste product. Then you have the dark reactions, where we use the stored redox energy and ATP to take carbon dioxide out of the air and convert it to a sugar. So that's a schematic structure. The light reactions produce ATP, and you produce reduced you produce an ATPH, which is fixed hydrogen, and then you use these two compounds to take CO2 out of the atmosphere in the Kelvin cycle and to produce the sugar. And the electron flow in the chloroplasts and cyanobacteria is that you have here first photosystem 2, where you have the water splitting, then you have you reduce plastoquinone to plastoquinone. This goes on to the cytochrome B6F complex. So it gets oxidized again, a very important step, because without oxidized plastoquinone, photosystem 2 cannot operate. And then you get electron transfer onto plastocyanine, onto photosystem 1, and the electron back further to NADPH, and at the end you generate ATP also, because you generate a pH gradient and an electric voltage, so-called membrane potential, across the photosynthetic membrane. And, but the major weak point is Rubisco, and because it cannot accurately discriminate between carbon dioxide and oxygen. So Rubisco incorporates carbon dioxide into ribulose 1,5-biphosphate. Then you have the splitting, and then you have a number of reactions when you regenerate the ribulose 5-phosphate. And here for synthesis, you use the NADPH, the fixed hydrogen, and you use the ATP. And the plant enzyme is very sluggish. There's only 3.3 reaction cycles per second. Catalase, for instance, has 10 million reaction cycles per second. And it's by far the most abundant soluble protein, which compensates for the sluggish activity. And as I said, it discriminates poorly between carbon dioxide and oxygen. You get an incorrect product, 2-phosphoglycolate. This is degraded using a lot of energy in the plant. And about 1/3 of the absorbed energy of the absorbed photons is used for the degradation of the phosphoglycolate. So for most plants, 4.5% is considered to be the upper limit of the efficiency of photosynthesis. But in reality, it's less than 1% of the energy of the sunlight, which is stored in the form of biomass. And if you go on about what is at the end the efficiency in biofuel production, then biofuels in German biodiesel, which is made from rapeseed, is 0.08%, practically nothing. If you go to Brazil and bioethanol, then it's 0.3% of the sun's energy, which is found in the ethanol. Palm oil diesel is 0.4%, so it's practically nothing. If you would use a photovoltaic cell, you would get 20%. And in addition, with the photovoltaic cell and the electric battery, you have an energy usage in the car by 90%. If you burn diesel, you have a 30%. 70% is wasted. Is wasted in that process, in burning the fuel, the gasoline, in the engine of the car. And so I would actually go for photovoltaic cells, put them somewhere here. And I read actually that Singapore had planned to transport sunlight, the energy by electric cables from Australia to Singapore, which I would like very much, but I learned yesterday that the plans were given up. I hope Singapore can revive these plans to transfer energy from Australia to Singapore. And so my vision how to use photosynthesis would be to improve the Rubisco, and you would be able to increase the yield of photosynthesis by 50% to 100%. One could try to expand the wavelength range of the plants, of the light which is absorbed. And we also can reduce the photoinhibition, which is a problem mainly in photosystem 2. And water availability will be a problem because you need a lot of water to produce the biofuel. We also can change the mechanism of carbon dioxide fixation in plants. The Kelvin cycle with Rubisco, as I said, is very inefficient. And for instance, there's archaea contain an acetyl-CoA carboxylase, which is the most efficient CO2 fixing enzyme. But this will take a lot of time. So what has been improved by attempts to, what has been achieved by attempts to improve photosynthesis, and what is already available is that you can degrade the incorrect product phosphoglycolate by different enzymes. And this leads already to a 50% increase in biomass. You can optimize the Kelvin cycle enzymes, and you get up to 60% increase in biomass. And also, when you increase the B6F complex concentration, which is the one which reduces the plastoquinol and which oxidizes the plastoquinol and gets the plastoquinol back, and so less photo damage at photosystem 2 occurs, and you will have an up to 70% increase in biomass. And what is nice, that these increases are additive. So you should be able to triple the amount of photosynthetic yield in the future. And I'm very optimistic, but I don't think we need that for biofuel production. I think we need that to feed the world's still growing population. That's, I think, why that is a very important thing. Now I change gears and I come to the major, to the suggested title of my talk. And so we talk about the great oxygenation event, also called the oxygen catastrophe or the oxygen crisis. And it was the most significant extinction event in Earth's history. And it was caused by the invention of the oxygenic photosynthesis by the ancestors of the present day's cyanobacteria. And if we have a look on the Earth's history, we learned yesterday that Earth exists since about 4.6 billion years, and life came into existence pretty early, but most likely deep in the oceans, so they had no problem with the UV light from the sun. And so photosynthesis already existed here. That was unoxygenic photosynthesis. Oxygenic photosynthesis probably about 3 billion years ago. And at the beginning didn't lead to an increase in the oxygen concentration because there was much reducing agents like iron 2 and sulfides available. They absorbed the oxygen. And only at about 2.5 billion years ago, we got an increase in the oxygen concentration in the atmosphere to about 1%. It stayed there for 2 more billion years. And then we got an increase to the 20%, which we have now, most likely to the colonization of the land by the plants. And why is oxygen toxic? And I show you here the reactive oxygen species. Here you have the triplet oxygen, which is what we have in the atmosphere. And by energy transfer, you can generate singlet oxygen, happens in photosynthesis and photosystem 2, which is very highly damaging there. You can generate the superoxide radical, highly toxic. A second electron, then you get the hydrogen peroxide. Third electron, you get a hydroxy radical and a water molecule. That's the most dangerous species, which you don't like to have in your body. And then putting a fourth electron, you get a second water molecule back. And you have to get rid of all these reactive oxygen species. And what can you do? Life could survive in oxygen-free niches. You can remove dangerous intermediates by superoxide dismutases or catalysis. And you also can remove molecular oxygen via reduction to water by the oxygen reductases. And nature has invented two membrane-integrated oxygen reductases. One is the heme copper containing terminal oxidases, which includes the cytochrome C oxidases of your own body. And we have the cytochrome BD oxidases, which oxidize quinols. And the BD oxidases don't pump protons. They are insensitive to sulfides, and they provide a survival advantage, for instance, in your gut when sulfides are present. And they are also essential enzymes for the pathogenicity of many human bacterial pathogens, like Mycobacterium tuberculosis, causing tuberculosis, and also some pathogenic E. coli also depend on the availability of the BD oxidases. So we determined the structures of various BD oxidases in our lab. The first one we did by X-ray crystallography, and it took us 12 years to solve the structure. Yeah? And it was 3 students in a row before Shara Safarian got the structure. Lousy resolution. Then we used now the cryo-electron microscopy. We determined the structures of the E. coli enzyme, that of Mycobacterium tuberculosis, and then the second enzyme of E. coli. And now we have one more enzyme. And this was done primarily by cryo-electron crystallography, which takes about 3 to 4 months after starting the project, so much, much faster. So we gave up X-ray crystallography. And now in the respiratory chain, we have the terminal enzyme, we have the cytochrome C oxidase, uses cytochrome C, and in order to reduce the oxygen to water here. And the heme copper terminal oxidase, which include the cytochrome C oxidases, of course have the reaction. We have oxygen, which comes from the membrane. We have 4 protons from the inner side of the mitochondria or bacteria. We have 4 cytochrome C molecules, reduced cytochrome C molecules, providing the electrons from the outer side. So we get generation of a electric voltage, so-called membrane potential across the membrane, and we also get a pH gradient across the membrane. And in addition, the heme copper terminal oxidases have invented a mechanism to pump 4 additional protons across the membrane, and this process is not well understood. So we still have to learn a lot about cytochrome C oxidase. We got the crystals, and for that we invented the trick of co-crystallization with antibody fragments. So we make first monoclonal antibodies against our targets, and then we produce the antibody fragments and make the complex, crystallize the complex. Also explained yesterday by Brian Kopelka. And so that's then the structure which we have. We have here the cytochrome C oxidase from our soil bacterium. And you have here the cytochrome C, which provides an electron first onto a kappa A. Electron jumps on to first to a heme A. Then it goes on to a kappa B, heme A3 complex here, which is actually the binding site for the oxygen at the heme A3, where it is reduced and water is formed. So this happens here. And so I'm now focusing on what happens in that part of the cytochrome C oxidase. So what is in this binuclear site? You have here the kappa B with some kappa ligands. We have here a covalently linked tyrosine here. And another group determined the structure of another cytochrome C oxidase. And they published peroxybridge seems to fit the end electron density here pretty well, but it's not expected in the oxidized enzyme. Our own work on our bacterium actually also showed that by far the best fit is a peroxide in between the heme A3 ion and the copper B. So here is what we think is a peroxide. And but this is not accepted. Now I should say a little bit about the catalytic cycle of cytochrome C oxidase. And actually what people believe is, of course, you have the oxidized state. You have an iron formally +3 and the copper formally +2. And then you get an electron onto it. You get so-called E state, electronated state. Then you put a second electron. You get so-called R state, where the iron and the copper are reduced. And this then leads binding to the oxygen. And here, then you form the so-called compound A. And the classical first idea was then you put the electrons from the iron and the copper onto the oxygen, you get a peroxide. And this is why this is called the P state, having that structure. And then put a third electron onto it, and you reduce most likely the copper. It's called the reduced P state. This converts to an oxoferryl, according to the classic view. And the oxoferryl then actually you put on the fourth electron and the proton, and you get a hydroxide bound to the iron. At the end, you come back to the O state. This is what most likely you see in many textbooks. A few years ago, due to Raman spectroscopy, there was hints that actually already here, in that so-called P state, you have already a splitting of the oxygen molecule. And for splitting the dioxygen, you need 4 electrons, and they can be provided. 2 of them can only be provided by the copper and the iron. One could be provided by the iron generated +4. And the fourth electron, there are ideas that you might get an iron +5. You might get you might alternatively you get a copper +3, or you might have a protein residue, most likely the tyrosine, which is part of the active site. And but also some people also investigate the idea that you have porphyrin radical, form of porphyrin radical, so the heme A3 itself donates the missing electron, and then you go on that way. So, and this is probably what used to be, still is the most generally accepted way for the catalytic cycle of cytochrome C oxidase. So we went to try to determine the structures by electron cryomicroscopy. So we generate the O state with our enzyme. We collect about 5,000 movies. We get 200,000 particles and get here the structure. Then we do the reduced state by chemical reduction, which is supposed to bind the oxygen. And then you get a P state. P state you can get with when you use carbon monoxide together with oxygen, you form the P state. And if you add an excess of hydrogen peroxide, you get the so-called F state. And we went on to analyze the structures of these cycle intermediates. So, so here you see the spectra of these compounds, and we got all the structures of all these 4 intermediates. And we could also improve resolution up to 1.9 angstrom resolution. And of course, we checked about the structures of the intermediate states, and we found low B factors at the binuclear centers, which means that the structure is pretty accurate around our binuclear center. And so what we also used with a density modification procedure. So this is the original electron the microscopic density. When you do the density modification, you can even see the holes in the density of the vinyl rings. So pretty good resolution. And what we also can see is now that we can clearly see the pathway of the oxygen molecules in the enzyme. You have the entrance towards this pathway is located here in the hydrophobic interior of the membrane. That is plausible because oxygen is enriched in membranes by a factor of 7. So the oxygen concentration in the lipid membranes is by a factor of 7 higher than in solution. And so that is here part of the pathway for the oxygen. And what is also very nice, here you have the entry site, you find a queue of oxygen molecules. So they align themselves and wait for being consumed at the active site. And even here, and this is actually here's the active site. That is the He3 iron, which binds the oxygen nearby carbon. And here that's the last oxygen of the queue. And when you look more in detail, you see the bridging peroxide, which is this green one here, which is located between the copper and the He3 iron here. So and this is then here the model, and now this is a different view. You can how these oxygen molecules lies in the active site, and this means that we have this peroxide bridge there in the oxidized form, which is not really accepted by many people. And in X-ray crystallography, the claim was that it's an artifact of reducing of the X-ray crystallography, because X-ray crystallographies also produce electrons, and you have oxygen around, and so it's an artifact, but it cannot be an artifact in the electron cryomicroscopy. And then we determine the structure of the reduced state, and what you see, no density in between the He3 iron and the carbon B, which used to be expected. One of these neighboring oxygen is replaced by a water molecule, and all this queue of water molecules is gone because we have reduced, chemically reduced the enzyme with dithionite. And the only significant difference with respect to the proteins that we have a side chain flipping of a lysine residue. So this is the lysine residue here, and this is is found here. That's in the oxidized form. It has that conformation, and if you put an electron then onto the binuclear side, you get a movement of the side chain closer to the upper side. And this most likely indicates that the lysine becomes protonated or is protonated, and then it moves closer to the to the electrons, which upon the reduction of the active site, and indicates that probably the lysine is a proton donor for the water formation there. Then we went for the P state, and the P state structure was the big surprise, because we did not see here the expected oxoferrile position. We saw instead of that, we see oxygen-like density between the He3 and the iron. So this is here the model here, and these are different views. So we have here this most likely dioxygen compound between the in the water molecule here between the carbon B and the iron, and that was probably the biggest surprise, because we expected this oxoferrile there. And so interpretation of that density could be a peroxide, could be a dioxide species, and actually, if we believe that in the binuclear center, in the O state, we have a peroxide, then in the P state we should have a neutral dioxygen, because the P state is converted to the O state by addition of 2 electrons. And so this is what was our preferred interpretation now. If we go on for the F state, we see here that we have actually a density which also for dioxygen molecule, or near the carbon B, bound to the carbon B now. And according to the expectation, the student originally modeled it as an oxoferrile, but this is not convincing. And so we did another modeling, and this is when you do the modeling for the absence of an oxoferrile. Problem here is that if you model here the oxygen atom bound to the iron, the distance here becomes too short, because you displace the iron atom to this direction, and this gets too short. And this now looks much nicer. And we have here that what we believe is a superoxide as a ligand to the carbon B, and there's a water molecule in here. And from inorganic chemistry, we learn that when a carbon +1 meets a dioxygen molecule, we get a carbon 2+ superoxide complex. So we suspect that this density here, there, is a superoxide. And we also have other evidence for that, because when you generate the F state, that is seen here in that part of the slide, absorbed slide at 582, and when you add catalase, then the F state actually is reduced, and you get this new state, and this new state looks very much like an O state. And we convert, by addition of catalase, we convert the F state to an O state. And this explanation is, and this is most easily explained by the presence of the superoxide in the F state. And if the superoxide takes an electron from cytochrome C oxidase, it leads also to the appearance of a tyrosine radical, located on tyrosine 164, that we know. And the peroxide, which is released from the enzyme, is split by catalase. And this then gives the optical feature of the oxidized form of cytochrome C oxidase with a ferric heme A3 ion. And so we have a most likely scenario for the second half of the catalytic cycle. The P state contains a neutral dioxygen, no oxoferryle, and it is an oxygenated oxidized state. And addition of one electron then leads to the F state. And upon this, the bound dioxygen is converted to a superoxide. And the F state does not contain an oxoferryle, but a ferric heme A3 iron. And if you add another electron, then you get the O state, and you have the bridging peroxide, so which makes complete sense. So we can phrase it differently, and this means the P state actually is an oxygenated O state, and the O state is the P state. So this sounds confusing. And so our present model of the cycle is that you have the R state here, iron +3, copper +2. You bind the oxygen here, you get the P state, then you get the electron, you get the F state, we can turn it to superoxide. Then you put on a second electron, you get a peroxide bridge, and then we have to go to the E state. That is, I think, the most dangerous step, because when you put an electron onto peroxide, you get the OH- radical, and you have to prevent it somehow. And we have to think how this is prevented. So we do some experiments, but our E state is not so clear for the moment. And one idea was, of course, maybe you have to rotate the catalytic cycle simply by 180 degrees and you are back. But this seems not to be true, because the E state is also no indication for an oxoferryle. So in order to get the E state, we treat the F state cytochrome C oxidase with carbon monoxide. Carbon monoxide is a 2-electron donor. But as I said, our cryo-M structure appears to exclude the present oxoferryle ion in the BNC, in the binuclear center. So it's not a 180 degree of the cycle. Of course, with that revolutionary proposal, we get a lot of criticism. Now we have to defend our model and go on. And I have to, of course, to acknowledge to the people. These are the people working on cytochrome C oxidase. Jürgen Köpke did the X-ray crystallography. Florian Hilbers did biosynthesis of the enzyme. Hao Xie did lots of chemistry, and it was Iris von der Hocht who discovered the conversion of the F state to an O state by addition of catalase. And for the intermediates of the catalytic cycle, it was mainly Felix Kolbe here, together with the help of Shara Safarian, who was also responsible for all our PD oxidase structures. Of course, we need a highly competent electron microscopic facility, which is now used by Sonja Welch. And originally, it was set up by Derek Mills, but he unfortunately died a few years ago. But we could close the gap with Sonja Welch. And without this know-how, our work would not have been possible. So that's the end of my talk, and I thank you for your attention.
Thank you. We now invite Prof Mulakaya to moderate the Q&A session.
Thank you very much, Professor Michel, for this great talk that first told us about the potential, the big picture of how one may be able to improve photosynthesis, and then demonstrated to us how getting this very detailed atomic knowledge of membrane complexes is actually able to translate into very, very precise mechanistic knowledge about life. So maybe I'll just take the first question. I know that you mentioned that it is amazing, right, that photovoltaics can be 20% efficient and plants are only maybe 1% efficient. But of course, there is this caveat that anyone can take a seed and throw it on a bit of dirt and put some water, and then you have an energy conversion machine of 1%. Anyone can do that, but if you want to achieve the 20%, then indeed you need an industry. You need to be able to produce a lot of photovoltaics. So do you believe that humans will be able to overcome the challenge to make photovoltaics actually a feasible option to generate all this amount of energy that we require?
Oh, yeah, that's already, the calculations have been done, and actually it's not a huge area which you need to generate all the electricity which is required in the world. Of course, in Europe there has been this Desertec initiative where you generate electricity not only by photovoltaics but also by mirrors focusing light onto tubes which are filled with some liquid, with salty liquid, which heat up to 400 centigrade, and then you, via steam, you generate electricity the classical way. And this has the advantage that you can store the heated liquid also during night, and so you get electricity generation also in the night. And this was called Desertec initiative, and it was supposed that you transfer the electricity via this high voltage high voltage cables to Central Europe and solve our energy problem. This has been given up, I don't know why. But actually now the British, they are trying to get a cable from Morocco to the UK, to the United Kingdom, about this 3,000 kilometers.
So you're optimistic?
I'm very optimistic that this will work out. And my idea is that you have to stay electric as long as you can, because each conversion into chemical compounds, you lose a lot of energy. So I don't believe that hydrogen is the big future, because you waste too much of the primary energy.
Okay. We'll take a question up there.
Yeah, thank you for giving your nice talk. My name is Dong-seok Kim from South Korea. I have a simple question about the chloroplast part. As you know that there are so many organisms having photosynthetic membrane, such as cyanobacteria and algae. So I was wondering, why did you choose the purple bacteria to analyze the structure of photosynthetic membrane?
Because they are very easy to grow, have a very simple composition, and these reaction centers, they are more stable than, for instance, photosystem 2 or photosystem 1 from the plants. It's much easier to get them. They are more stable. And for the system 2, we have also heterogeneity and instability. Actually, I thought when I solved the purple reaction center that I should be able to work out the structure of photosystem 2 as an emeritus, after retirement. But actually, the speed the field developed was much, much faster than I expected. And photosystem 2 was solved beginning of the 2000s.
Thank you.
All right, so we have a question online, which I actually like. It's what is your view on the supercomplexes? Do you think they are real? If so, what could be the advantages of forming them in the cell? And this question is by Sahutsai Inwongwan from Thailand. It refers to observations of these complexes in the membranes that appear to form, to be in spatial proximity.
The supercomplexes are real. It's no doubt.
Real. Okay.
Real. And if we have the supercomplexes, we have a pretty short distance for the diffusion of the quinoles, and also cytochrome C doesn't have to move move a lot.
Yeah.
But actually, it's not absolutely required. There are investigations by spectroscopy telling you that still complexes are pretty mobile.
Is there a data
But they are there.
you can mutate to stop formation of supercomplexes? Has that been looked at?
Quite often you need cardiolipin for also, the cardiolipin in order to get these kind of supercomplexes. And so without cardiolipin, you don't get much of these supercomplexes. And in some examples, you have the cytochrome C covalently bound to the B6F complex, to the BC1 complex.
Yeah.
So this cannot diffuse over low distance. So the cytochrome C oxidase and the BC1 complex has to be close together if you have the covalent attachment of cytochrome C to the BC1 complex.
We will take another question up here.
Hiya, this is Tim from New Zealand. Thank you for the detailed explanation about oxygen pathway and mechanism for using this enzyme to catalyze oxygen reactions. So considering the high promotion of hydrogen fuel cell cars nowadays, so I'm just wondering if you, and a lot of researchers are carrying out research about using this kind of complex or enzyme for electrocatalysis to enhance the oxygen reduction reaction. So, which is an important part for hydrogen fuel cell. So I'm just wondering if you have any comment about the future possibility of using this this kind of complex for electrocatalysis.
I don't think that biology proteins can help you much because they are not so stable. So the inorganic materials are much more stable and much better suited for this kind of applications. Yeah?
Okay, cool.
Yeah? So of course you need this proton conducting membrane, things like this.
Yeah.
And biology cannot compete.
Yeah, understand that.
Unfortunately, I would like that our work in biology can help to solve these problems, but I'm not optimistic there.
Yeah, that's right. I just feel like considering like my own experience, I found the efficiency is like quite low and the stability is quite poor. So just wondering, just curious about your comment about it. Thank you very much.
Thank you.
We will take a question up there.
Hi, I'm Wanita, a PhD candidate in plant science. I was wondering that how can you see in the future to grow these kind of modified plants, since we try to adjust or try to improve the abilities of the enzymes, but still the plant needs another factors like fertilizers or water to grow the plant. So do you expect to see this kind of plant growing around everywhere on the earth, or how do you think about that?
Of course, you have to analyze what is the limitation for the growth plants or the growth of the algae. And in reality, we have, I think, two major problems. One is water. You have to have sufficient water. And the other point is that most often you need to have a limitation of nitrogen. Yeah, so you have to add nitrogen. And I don't think it's a good idea to convert the plants into nitrogen fixing, and to have, because the yield of nitrogen fixation is low, and you waste a lot of energy. And if you look around, what is the biomass production of nitrogen fixing plants? This means, of course, it's the bacteria in the nodules, in the roots, which produce the plants, is pretty low, and the yield of biomass nitrogen fixers is not good. Yeah?
Thank you very much. So we have now run out of time, unfortunately, for this session. So thank you very much, Prof. Michel, for your seminar.
Thank you.
Thank you, Prof. Michel, and thank you, Prof. Mulakaya. Please now welcome Sir David MacMillan, who will talk about the development of asymmetric organocatalysis and metallaphotoredox. The Q&A session will be moderated by Prof. Sergei Kozlov. Sir David, please.
Thank you. Good morning. Hopefully there's my slides. It's great to be here to see this very enthusiastic, diverse audience. It's a great chance for me to actually get to be here, to be part of this conference for the first time. Before I begin, I was going to start off by telling you a little bit about me, who I am. I know this is an audience that comes from a lot of different backgrounds. As you might be able to tell from my accent, I'm from Scotland. Here's a map of where Scotland is, and I hope this is not insulting to you, but most people in America have no idea where Scotland is. So I always like to sort of show this on a map. This is exactly where we are. For those of you who don't know where Scotland is, there's a really easy mnemonic. There's a really easy way to remember it. It's always on top of England. Okay, so it's really, really, really simple. All right, however, where am I from? I'm from just outside of Glasgow, this tiny steelworking town. My father was a steelworker, my mother was a maid. And this is where I grew up, and this is the home in which I grew up. And I wanted to show this because I think this is also true of a lot of places in Singapore. I grew up in what's called a council house. A council house is where you rent the house from the government, and that's exactly where we grew up. And while we were working class and grew up being working class, we were an extremely happy family. So I always tell people we were working class, but we were very wealthy people because we really enjoyed ourselves every single day. I love this photograph because you can tell it's easily the 1970s because of the wallpaper and the curtains are pretty unique. All right, now I was born in 1968, and you might ask yourself why I would show you these photographs. But after I won the Nobel Prize, the Nobel Foundation contacted me and said, can you send us photographs of you as a baby? And I was very confused by this, and I said, why? Why would I do this? And they said, well, we want to put them on our website. I'm like, okay. So I didn't have any photographs of me as a baby, so I contacted my sister back in Scotland, and she sent me these. These are now on the Nobel Prize website. And I think this is the greatest practical joke of all time, because I'm pretty sure that's not me. I'm pretty sure that's my sister, actually, who's very proud of the fact that she's now on the Nobel Prize website. Okay, so when I was about 12 years old, my brother decided to do a pretty remarkable thing, and he decided he wanted to go to university, to college. This was very controversial in my family because we didn't know anyone who'd ever gone to university before. But he decided he wanted to go, and the day he graduated, he actually got a job that paid more money than my father, who was a steelworker. So immediately, I was told I had also to go to university so that I could make as much money as my father. So the decision to go to university wasn't really my own, to be honest. It was really those of my parents. But I was very fortunate that my brother decided to do this. Now, he received a bachelor's degree in physics, so I was told I had to go to university to do physics, and off I went to Glasgow to do that. But after about 6 months, I realized I really didn't like physics, and I wasn't very good at it. So I decided I had to make a change, and I decided to move over to chemistry. I really fell in love with organic chemistry, which I'm going to tell you about today. And this is actually my graduating class. It was an amazing experience for me because going to university, I got to meet so many different people from so many different backgrounds. And as much as an education in science, it was an education in sociology and meeting people, which was one of the most important things about going to college. So after doing this, I decided I had to go off and do a PhD. I was in love with the United States because I watched NFL and I watched a lot of independent movies and I liked a lot of groups that came from America. It really wasn't anything to do with the science, it was everything to do with moving to America. So I decided to use my degree as a passport to move to the States. Now, I applied to 19 different universities in America. I wrote to 19 different people, and only one person responded, and it was basically this gentleman I'll show you here. This is Hal Moore, and it was very fortunate that he did, because he said to me, you cannot get a PhD in America simply by sending a letter. He said you have to fill out an application form, and that's exactly what I did. And I always love to tell this story about how much fortune there is along in everyone's lives as you move through your academic career. And this was very good fortune for me that he took the time to respond to me and actually send me this application form. So off I went to California, and that's where I began my PhD in 1990. Okay, I'm going to come back to this later in my talk. That was me in a jacuzzi, by the way. I thought I'd taken that slide out, but anyway, it doesn't really matter. But 2021 is when I received the Nobel Prize, and I received the Nobel Prize along with Ben List. And since receiving it, I've been asked an awful lot of questions, but probably the number one question I've been asked is, asymmetric organocatalysis, what on earth does this actually mean? So what I thought I'd do for you today is sort of break this down into its individual components. First of all, what is catalysis? I think most people here probably know what catalysis is, but one of my favorite ways to talk about this with either high school students or undergraduate students is to first of all point out that every single thing in this room, every material, every substance, every single thing in my office, which is shown here, is made from a chemical reaction. And here are just some of those materials, and it's flashing through those different chemical reactions. And if I focus in on one of them, which is my coffee cup, which is shown right here, and if you look at this is the chemical reaction to make caffeine the key constituent in my coffee. And as it happens, most chemical reactions require energy. Most of them are not spontaneous. And the way that we teach this to incoming chemistry students is we point out, we use these energy diagrams. And again, I don't try to make this too technical for people, but I always point out the way to think about these energy diagrams is, imagine every night when you're walking home, you have to walk over a large hill. That would require a lot of energy. If, however, somehow you're able to build a tunnel through that hill, that would make it much easier to actually get home every night. And that's exactly what catalysis does for chemical reactions. It actually makes reactions easier, faster, and probably more important, it actually allows chemical reactions to happen that were previously impossible, which obviously has very large implications. Now, if you're wondering what is the impact of catalysis on the world, there's a variety of good ways to sort of demonstrate this. I'm going to show this with, first of all, just one chemical reaction. This is basically the population of the Earth over the last 2,000 years, and you can see it's pretty stable. Until the beginning of the 20th century, there's an inflection point, and then it takes off to over 8 billion people. As it happens, you couldn't have 8 billion people on this planet without one catalytic reaction, the conversion of a nitrogen over to ammonia. Why is that? Well, the reason is we need ammonia to make food, and without this catalytic reaction, you simply couldn't have 8 billion people. One of my favorite statistics associated with this is the fact if you think about your body right now, you think about all the protein, the DNA, tissues, your organs, and all those nitrogen atoms that are in your body right now, 50% of them actually come from a factory in Germany. That's an amazing statistic from one catalytic reaction. Okay, now 90% of industrial scale chemical reactions use catalysis. 35% of the world's GDP is based upon catalysis. And as we think about solutions to the problems that we're going to face on Earth over the next few decades, that number is only going to increase. I'll just flash through some other ways in which catalysis is important for society here. I'm not going to go through them all, but I hope you can sort of agree that catalysis is critical for societal impact. Okay, so that's catalysis. Next question, what about asymmetric? What does asymmetric mean? Well, this, as it happens, is really easy to explain to people who don't do this type of chemistry. For example, look at your hands and ask yourself, are your hands identical to each other? They kind of look like they're identical, but it turns out they're not. They're not because they're mirror images of each other that are not superimposable on each other. How can I prove that to you? Well, for example, if I was to take the glove for my left hand, we know that it will nicely fit on your left hand, but if you take that same glove and try and put it on your right hand, it doesn't work. The right hand glove does not recognize the left hand, and vice versa. Okay, now why is that important? Why do we care about mirror images? Well, this same phenomenon happens with organic molecules. So these are two molecules that are mirror images, but you cannot superimpose them on each other. And one of the most interesting aspects of this, if you go into a lab and try and differentiate these molecules, it's actually very difficult. It involves pretty expensive instrumentation. This would take about 20 to 25 minutes of time just to tell those molecules apart. However, if I was to take, this is my daughter, Emma, when she was 3 years old, back when she was a really nice person. And basically, even as a 3-year-old, she could even tell these molecules apart instantly simply by smelling them. So why is that? Why is it your body can tell them apart instantly? Well, it's because your body is made up with effectively one mirror image and not the other mirror image. And as much as the way that your right-hand glove can recognize your right hand, but it can't recognize your left hand, your body recognizes these molecules in different ways. So as that happens, that has lots of really interesting implications. First and foremost of which is the pharmaceutical industry. As you might expect, it becomes really important to be able to make one mirror image of a drug molecule, but not the other one. The other one is undesirable. It can be dangerous, it can be toxic, they can give you side effects. So how are we going to make this one mirror image? Well, we're going to take effectively catalysis, and so now this becomes known as asymmetric catalysis to make one without making the other one. Okay, so far so good. Next question becomes, what does organo mean? Okay, so I'm going to take you now back to 1996, and if you ask yourself, how could we perform asymmetric catalysis in 1996, there was 2 ways you could do it. The first way was to use biocatalysis, to use the enzymes of life, or from animals or from humans to do this. And the second way was human-made, and this was based upon using metals which have been designed for the purpose to allow you to make one mirror image and not make the other one. So why 1996? Well, 1996 is when I finished my PhD, working for this fantastic gentleman, Larry Overman. And upon completing my PhD at Irvine, I actually moved back across America to Harvard, and I went to join the labs of Professor Dave Evans. Dave, who unfortunately passed away last year, is a genius of our field. He's one of the, I would say, foundational scientists of the field of organic chemistry. And one of the things he absolutely was is a master in this area of asymmetric catalysis using metals. And here are some of the metals that Dave and his group worked on. And I joined his lab to learn about doing exactly this. And it was a really fantastic time. However, during that time, every single day, I'd be involved with this contraption. So most of you probably know what this is. This is a glove box. It's a glove box that has a different environment in the inside from our normal outside environment. Why is this? Well, it's because many metals are not suitable or could not be stable in our outside environment, so you have to keep them under argon or nitrogen for them to be stable. And after 2 years of working in this glove box, 8 hours a day, I started to ask the following question: why are we spending so much time in a glove box every day? This just seems like unnatural as chemists that we have to do this. So with this in mind, you can actually look at a typical catalyst that would be used at that time, and even visually, you can see you can break it right down the middle, and it's what looks like 2 different parts. On the right-hand side is the metal center, and metals, in certain cases, but not all, but in certain cases can be expensive, they can be toxic, they can be difficult to work with, and there's also sustainability issues. For example, right now, we only have enough palladium on our planet to last 90 years, which is also a really sort of scary number. At the same time, if you look at the left-hand side, this is the organic molecules. These are inexpensive, they're safe, they're sustainable, they're broadly available, and after you use them, they go right back into the life cycle, return back to us in a relatively rapid time frame. And as such, we can ask ourselves, what if we only use the organic part as the catalyst? And this eventually became known as organocatalysis. Okay, so 1998, I landed a job as an assistant professor over at Berkeley, and this is actually one of the first photographs I ever took of my group. I love this photograph. It's 10 past 10 on a Friday night, and you can see this young group in their first year, working hard, trying to make a difference, trying to have an impact. And you might ask yourself, well, what did we want to work on? Well, we decided we wanted to work on organocatalysis, because we started to realize that this idea of using these types of catalysis would have all these advantages. They should be able to be available from nature's building blocks. They should not be sensitive to air and water, the same way we're not sensitive to air and water. They should be inexpensive, they're broadly available. You wouldn't have to work in these contraptions of these glove boxes. And they should be sustainable, right? They should be non-toxic, they should be recyclable. And all of these things seem like sort of great aspects to this, but this was not the reason why I was mainly interested in doing this. The main reason was the concept, could we actually develop a way of using these organic catalysis or organic catalysts, not to perform one transformation or one chemical reaction, but to perform hundreds. Now, this was a pretty grandiose idea because at the time, I had absolutely no idea how to do that, zero. So that was just wishful thinking at the time. However, in my first year, I was fortunate to have great graduate students. This is one of them, Tristan Lambert, who's now a professor at Cornell. And he actually came up to me one day and said, what is the mechanism of reductive amination? It's a pretty simple mechanism. As a young first-year professor, I ran to the board to explain this to him. And I said, you take an aldehyde, you take an amine, it reversibly forms an aminiamine, and it's only when you trap the hydride it's possible when it has this electronic configuration. It's basically activated to do that. And it was right there, right then, I had this sort of quintessential eureka moment, because I said, wait a minute, if you think about this, if you take an alpha-beta unsaturated aldehyde and amine, that should reversibly form an aminiamine, and that looks awfully like a field of catalysis that uses metals. So if I show that in a slightly more conventional format, in this top case, using metals for what's called LUMO-lowering catalysis has been used for literally hundreds of reactions. The bottom case has been used for basically zero at that time. But if these are simultaneous equations, that means you should be able to use this organic catalyst to effectively perform, hopefully, hundreds of reactions. So at that moment, we decided we had to test this, and we had to pick a chemical reaction to test it with. And we decided to focus on the Diels-Alder reaction. Why the Diels-Alder? Well, Diels-Alder deservedly won the Nobel Prize in the 1950s for allowing you to take relatively simple start materials and with great predictability make relatively complex molecules you could use for drugs, for materials, etc. So this looked like a great sort of benchmark reaction to employ. And this notebook page is exactly the first time we actually ever performed this. Those are the two start material. Those are organic catalyst over the arrow. And if I scroll through this and go to the bottom, the result here, I hope you can see that, it says not racemic. Not racemic. What does that mean? Not racemic means it makes one mirror image in preference to the other one. So this was extraordinarily exciting. I remember basically leaving the lab, going into my office, closing the door, closing the blinds, and then I jumped up and down for about 5 minutes. And I called up my wife at the time and says, you won't believe this, but I think we're going to get tenure. And at the time, tenure was an incredibly important thing to an assistant professor. It was probably the =1 thing we were thinking about, even more than the reaction itself. But when my feet got back to the ground, I sort of realized that this number, this meant 48% EE, this meant a 48% excess of one mirror image over the other one. And if the chemistry community was going to take this seriously, this number had to be about 90%. So we're faced with a dilemma. Would we publish this, or would we go for the gusto and try and get a catalyst that would give us that 90% number? We decided to do the latter, and eventually we came upon this catalyst. And I really love telling this part of the story, because this catalyst is based upon phenylalanine, a building block of life, which is combined with acetone. Acetone is paint stripper. So you can see how inexpensive and available this catalyst would actually be. But more importantly, when we actually first employed it, you can see the transformation now gets to this 90% EE number. So that was really, again, very exciting. Okay, so at this stage, you have to publish this paper, you have to announce it to the world. And as a young assistant professor at the time, I always sort of get a chuckle when I sort of look at this first paper, because you can see the sort of excitement that we have in this paper. The first thing is we came up with a name for this area. We called it organocatalysis. And you might ask yourself, well, what's in a name? Is a name important? But I can tell you that naming things or vocabulary around science is extraordinarily important. So for example, Jean-Jacob Berzelius, who's a Swedish scientist, chemist, he actually came up with the term catalysis, as well as protein, polymer, isomer, organic, inorganic. He named all of these things. And the importance of naming things for the field, the vocabulary, has carried forward into the modern era. Directed evolution, machine learning, AI, nanotechnology, organocatalysis, these are all words that help you define fields and in many ways actually help you draw in other scientists. But more importantly, in this first paper, we introduced the concept of a generic activation mode. And you might wonder, what is a generic activation mode? But that's just simply a technical way of saying this should work for hundreds of reactions instead of one reaction. Now, the problem with that statement and putting it in our first paper, we'd actually only performed one reaction. We hadn't actually performed any more, so this was a little bit of a bold statement to begin with. And when we went off to test it, we went bang, bang, and it stopped. We got 3 reactions. We told the world we're going to do hundreds, we got 3 reactions. So again, this was a pretty scary moment. We had to go back and sort of think about the catalysts involved. And when we did so, I was really fortunate to have 2 fantastic young graduate students by the name of Joel Austin and Chris Bortz, who basically performed what I would call precision molecular engineering. What I mean by that is they effectively took away 2 carbons here and introduced 4 carbons there in a slightly different shape. And they stated that they felt this should be a general catalyst, meaning it should work for many reactions. Now, I call this precision engineering, and whenever I give talk to non-scientists, I always try and sort of convey what does this mean to in other ways of looking at the world. And the way I always do this, because I'm a big football fan, I always say this is very similar to, for example, precision athleticism, which give amazing outcomes. And this is Zlatan Ibrahimovic, a very famous superstar football player. And I always do this by showing this following goal. So this is this goal. It is this overhead kick. It's incredibly precise, just a fantastic goal, just an amazing goal. Okay, now many people have criticized me. I actually put this in my Nobel lecture, and many people sort of criticized me and said, yeah, you only put this in because this goal was scored against England. And I think that's actually really, really unfair. But let's watch it again. All right, so this is actually an amazing goal if you think about it. So, actually last year, Zlatan Ibrahimovic gave me a uniform that he signed for me, so it turns out it was fantastic. Okay, but the bigger point here is, can you actually use this precision engineering to have an impact? And so this was our original catalyst. It gave us 3 reactions. When we move now to the 2nd generation catalyst, it literally led to hundreds of reactions which are now successful. And in fact, Jorgensen and Hayashi came along, and they were able to show that using this other catalyst, you could expand this even further, which was really, really wonderful to see. Okay, so what different aspects of this become important? Well, one of the things that we realized, what we had performed here was a thing called iminium catalysis. There was another area called enamine catalysis, which Ben List, the co-recipient, received the prize for. And we started to think is there ways that you could start to combine these types of catalysis together? You might ask yourself, why would you want to do that? Why would you want to have 2 different catalysts going on at the same time? But you realize that nature does this all the time. Nature has these biocatalytic cascades that all beautifully sequence together to generate complexity from incredibly simple starting materials. So we said, well, enzymes are organic molecules. Maybe we can take these small organic molecules and do exactly the same thing. So this was the idea. Could we take these relatively simple starting materials, and could we now start to sequence them in multiple catalytic reactions? And the hope here is that we're going to generate a much more complex molecule than that we actually began with. And when we did it, it turns out it worked. And it worked beautifully. And it became an area in and of itself called cascade catalysis. And so whereas many years ago, you typically see 1 catalytic reaction in any given chemical reactions, now you see ones that have 5 or 6 different catalytic reactions all going on in a reaction vessel. Nature's known how to do this for forever, but we are now, as a community, figuring out how to do this to increase efficiency. In this case, we actually used this to make strychnine. This was the fastest known synthesis of strychnine, which you probably know as rat poison. You're probably wondering, why did we want to make rat poison? Well, strychnine, for organic chemists, is a very famous benchmark, which people use to sort of evaluate their technologies in terms of how quickly you can make it. And this was the fastest known asymmetric synthesis of it. So what other reactivity can you get from organocatalysis? And what we've been able to do, we're very excited to do, was to actually start to think about, could we come up with other types of catalysis to merge this with? And one that was really exciting to us was, could you actually start to take the principles of harnessing basically solar energy, so using visible light. inorganic chemists over the last 5 decades have really done a phenomenal job in thinking about how to capture solar energy to try and power the planet. And they've developed many catalysts that allow you to capture that energy. And along the way have delivered some really fantastic fundamental concepts. And we started to think, could we take those concepts and now move them into organic chemistry? Now with the idea of thinking about, can you accelerate the production of drugs or materials and so on and so forth. And this led to the development of photoredox catalysis in organic chemistry, and in and of itself has really become an area which is as large as organocatalysis, and also makes for some really pretty videos along the way, as you can see here. So, but we were really fortunate because this field of organocatalysis, which began, really led, at least in our lab, to the development of photoredox catalysis, which was something, again, which is really valuable, we felt, and important to be able to see. Okay, so the other major question we're often asked are, what are the applications of this type of organocatalysis? And I'll show you 3 that we pretty much thought of early on, and I'll show you 1 that we didn't actually see coming. The first one is for flavors and fragrances. And you might wonder, fragrances, does anyone really care about fragrances? But if you think about the volume of fragrance that's actually produced on the planet Earth every single day, it's absolutely enormous. And if you can change the scale, the way that you go about making those, that can actually have a reasonable impact. So Firmenich, which is a Swiss company, they actually worked with us towards the idea of, could we use this to make a perfume they make called Lily of the Valley? Previously, this had been made on 6 chemical steps, and we could now actually make it in 1 chemical step, now from using basically biomass and combining organocatalysis with this photocatalysis to do it in 1 chemical step. And in fact, Firmenich now in northern India make a variety of rose perfumes, which on I think it's 400 to 800 ton scale, using organocatalysis as well. One other way that people have sort of thought about, I'd say one prominent way that people have now really used this, though, has been for the pharmaceutical industry. The pharmaceutical industry has heavily adopted organocatalysis, not just for discovery of new drugs, but actually for the manufacture of them. This is Talcegipan. I think it's 5% of the world's population basically suffers from migraine. And what was exciting about this was Merck actually were able to make this single mirror image by using exactly the technology which I talked about at the beginning of my talk. About 2 months ago, I was over at Merck, and they actually showed me that they've now a new cancer treatment that's coming online, which actually uses 2 organocatalysis processes in combination with 2 biocatalysis concepts to drive the production of that molecule, which was really beautiful to see these various branches of asymmetric catalysis coming together to actually make some of these molecules. Okay, now the part we did not see coming, however, was this idea of democratizing catalysis. What does that mean? Well, it basically means that organocatalysis is extraordinarily inexpensive. It's very, very cheap based upon the materials you actually use to make it. And as a result of which, it's now basically used to teach asymmetric catalysis all over the world, as well as allows you to drive research in all continents around the world. And one of my favorite parts of this is it turns out it allows you to do asymmetric catalysis regardless of the resources that you basically have available to you. And people often ask me, what do you think is going to be the next big thing in organocatalysis? And I always say, I actually don't know. But one thing is it won't be based upon who has the most funding, the most money. It'll be based upon who has the best ideas. And for someone like myself, that really aligns well with my values, and that's why I was really proud of the fact it's been able to achieve this way or allow people to start thinking about asymmetric catalysis in this context. So what does the future hold for organocatalysis? Well, I think one aspect of this, which is very important, we're obviously moving towards 8 billion people. We have now reached 8 billion people. We're going to keep increasing. And we have to think about even adopting other ways of continuing to achieve sustainable catalysis. And these are things such as organocatalysis, biocatalysis, photocatalysis, electrocatalysis. But while I started out telling you that we really wanted to avoid metals, there's also a big chapter of this that's going to involve using base metals. Metals that are widely available, and can we actually teach those to effectively function the way that precious metals do? So that's going to be very critical for our planet as well. All right, so I'll finish off there. Oh, I just realized that I think someone's launched the wrong talk. See if I can exit out here and get to the, yeah. Fortunately, I have the talk here. And I basically just want to finish off, but I think it's an important point I want to finish with. I want to finish by thanking my family. I think we're all, as scientists, we do this because of our families. This is us dancing on basically on the Harbour Bridge in Sydney, down in Australia. And one of the things that I've learned in academia is amazing because it lets you work with fantastic people, allows you to work on your own ideas, but it also allows all of us to travel the world and get to connect with each other, which is obviously what we're doing today. So I wanted to sort of point out to the audience, and this is beyond the talk, but academia really is a fantastic profession. And I hope that people sort of understand how important it is that we continue to get people like yourself to be part of it. I also want to point out that after I received the Nobel Prize, we took the award money and we created a foundation. And there was a foundation to basically give all the money to charitable causes that will allow underprivileged students get to college. This is something that's very close to my own heart. But it's also something I think is really important right now, that we do care about people who don't have opportunities. What you do with your opportunities. Thank you very much. I was actually just going to say, what you do with your opportunities, everyone knows it's up to you what you do with your opportunities, but getting opportunities is the hardest part. And then finally, I just wanted to say, getting the opportunity to talk to people like yourself, and one of the things about the Nobel Prize that's been amazing, for me at least, is, and through Zoom, you can talk to people all over the world, and I've been able to sort of talk to lots of different high school students over the last year and a half, and that's been utterly fantastic. But one group that I got to talk to, and I think all the Nobels were involved with this, was the idea that the Ukrainian ambassador to the US contacted me and said, would I talk to displaced Ukrainian students? And I said yes, but I realized that I really couldn't imagine or identify with what these people were going through. So I was extraordinarily nervous to sort of give this talk. But when I talked to them, the thing that absolutely blew me away, it was completely profound, was the fact that how excited they were about science and talking to me and how jazzed they were. And for myself, imagining what they were going through, the fact that they were so excited about science is one of the sort of most, to me, heartwarming things I could ever imagine. So I don't know if my talk had any impact on them, but it had a massive, massive impact on me. So I hope I can answer some of your questions at the end of this, and I just want to finish off by thanking you very much. Thanks a lot.
May we now invite Prof Kozlov to moderate the Q&A session.
Yeah, please have a seat. So, thank you for a very inspiring talk, Professor MacMillan. So now we have a time for a few questions. And I see there is a gentleman there, so maybe you could start with the first question.
Yes, good morning, sir. Thank you very much for your wonderful talk. I'm also doing organic synthesis when I was doing my PhD. And what makes organocatalysis very fascinating to me is that the catalysis itself looks like a substrate. Yeah, because they have functional groups that could also somewhat interfere with the reaction. So when you're trying to develop or trying to choose organocatalysts, what makes a good candidate for let's say how do you choose which type of organocatalyst you're using, especially if the structure becomes more complex?
Yeah, it's a very good question. One of the parts about this which was really eye-opening to me, and if I refer you back to the part where we did this cascade catalysis and where you can now have 4 or 5 different catalysts in the same vessel, when we first started out, we thought that would actually be almost impossible because we assumed all of these things would simply all interact with the same things. But obviously chemistry is far more complex than that, and when we actually tested it, they didn't. So you started to build up this picture of the electronics involved and what was actually going to be lumilowering, homoraising. So you realize it's all like biology based on equilibria that's going on, and as long as you can control the electronics, it actually turns out to be far more straightforward than you would have ever imagined. So as long as you have a reasonable appreciation for the electronics of your catalyst versus each other and the other molecules you're working with, it's actually much more predictable and much more straightforward to design at the beginning.
Follow-up question. Would you say a computational type of development, or let's say AI, with the machine learning in trying to model the molecule with the electron density would be the somewhat future of this field?
Yeah, I mean, potentially. I mean, I think we're at this inflection point with AI where we now think it's going to solve all of the world's problems tomorrow and we don't have to do any thinking. I think that AI is going to be extraordinarily important. Right now, AI, the way it's being used in chemistry, the way most of it's used is for optimization, which is really important. But personally, I don't find that as intellectually interesting. But one of the things I do think which is really interesting with AI is, can AI help you ask questions? Because if you think about it, AI can only do what humans ask it to do. But if you can actually think about using ways for AI to question what are the reactions we should be trying to invent based upon the resources we have on the planet right now, I think that's a more interesting question. And then either through human invention or AI development, you could start to address some of those problems. So I think for me, that's where I see more excitement around AI. And I've talked to Demi at DeepMind about this, and there's a lot of interest in sort of moving forward with how do you think about the questions as much as you think about the solutions.
Thank you very much.
Thank you. This was a very important question and a great answer, of course. So I see there is another question from the audience, so please go ahead.
Thank you for very interesting presentation. My name is Gochapan Danganpai from Thailand. So just a question from your bachelor, because first you entrance to the physics and you move to the chemistry. What is the inspiration or motivation that push you to study about the organic chemistry?
Yeah, and I think everyone has different experiences in this regard. For me, and this is one part I always try to tell people, is you have to find your own sort of passion in life, and so when I started doing physics, I realized that I really, while it's an incredibly interesting area, it wasn't really good for me. I wasn't enjoying it, and I don't think my brain is abstract enough to actually be successful in physics. But my brother did, and he loved it, but I realized I was doing it because my brother did it. And so along the way, I sort of figured out I had to find what I like. And so I think it was really important to sort of sample lots of things and try lots of things, and then find the part that to you goes, wow, that's cool. I really like this. And for me, that was organic chemistry. So even other parts of chemistry I wasn't particularly good at or excited about, but organic made an awful lot of sense to me. And I think that for me was a lesson in life that when you find those sort of passions, it's good to sort of go in those directions if you can, if you're afforded those chances. So ultimately, the take home for me was like just finding what you're excited about and following that instinct is really, really important.
Thank you very much.
Thank you. This was a nice question. And before we go to the next question, let me ask one question asked online. So the question is, do you think organocatalysts will mostly replace metal-based catalysts in the future?
No, not at all. So this is a question you get all the time, and I think as sometimes as humans, maybe not as scientists, but we sort of get into these binary situations of yes, no, and can we replace everything with one thing, or can we replace, and so the question is organocatalysis better than metal catalysis, better than biocatalysis? And what you realize along the way is what you're doing is you're sort of developing really, hopefully, valuable tools for being able to address almost any problem. So from my perspective, organocatalysis is extraordinarily useful for a whole range of chemical reactions. There's a whole range of chemical reactions it's just not useful for, and those are ones that metals will be uniquely valuable at doing. So I think what we have to do is be able to understand and be comfortable with building up areas that are useful and continuing to think about new areas of catalysis as well. If you think about all the societal issues that we're facing as we move forward, those will require not just new catalysts, but new areas of catalysis if we're going to be successful. You don't want to sort of sit or rest on our laurels of what we have right now. We have to actually think about new forms of reactivity.
Oh, thank you. That's very important. Okay, so maybe we can proceed with the next question from the audience, please.
Thank you for a very clear and engaging talk. You've described an eureka moment in your career where you were able to about the similarity between organic compounds and metal-based catalysts. My question is, what do you think made you spot that similarity, and what prevented other scientists in the field from spotting that?
Yeah, I mean, I think that's a question that a lot of Nobel winners get asked, actually. What made you see something that other people didn't see? And we're exactly the wrong people to ask, because we don't know why we saw it. We saw what we saw. And I think it was along the way, probably, my guess is, it was, in our case, it was something that maybe other people thought was too obvious, that it was too simple. Maybe this is, or if that would work, someone would have found it by now, sort of thing. At least from our perspective, we looked at it and thought, that looks so unbelievably similar that we had to examine that really quickly. I mean, it would be crazy not to. And I always say to my group, that's an experiment that's too easy not to try. And so you have to do it. But knowing why no one or other people didn't do it is a tough question, because obviously we were able to spot it.
Thank you. And we have another question asked online about the impact of scientific work. So the question is, did you deliberately work on your science communication skills, or are they a product of experience and personality? And maybe you can comment on the importance of scientific communication.
Yeah, I mean, it's kind of funny, you know, a lot of people say to me a lot of the time, you're decent with scientific communication, and that's helpful. It's kind of funny, I actually look at young people nowadays, and young people are spectacular at communication. When I watch my group, and I watch probably the vast majority of people in this room give talks, they're amazing, and it's clearly become the case that you have to be good at, your science has to be good, but given all the noise which is out there of people talking about, and the chatter about what they're doing, you have to have good scientific communication skills if people are going to hear what you're doing. It's one thing to do something that's valuable, it's another thing for people to actually know about it and care about it, and I think communication skills becomes really important. But I do think this age group which is in front of me and the group I work with, the communication aspects of it have really been elevated enormously. I do think it's important. I think, you know, to be able to take an audience along is very critical. I grew up in Scotland, and Scotland, you know, your entertainment would be you'd go down the pub and you'd drink beer and you'd tell each other jokes. That's what you do. And so you become each other's form of entertainment. So you have to learn to be able to communicate with each other. That's what you do. You have to take the audience with you to a certain extent. So yeah, I think it's a function of where I grew up as much as anything.
Oh, thank you for this answer. And we have another question from the audience. Please, go ahead.
Hi, this is Zainab, and I'm from UCSI University. I would like to ask us as young scientists, sometimes during our research, we find a method and we expect a good result from this method. And then after implementing it, we face failure, or we face shortage of results. So what I want to ask is, through your study or experience, you were expecting more than 100 reactions, right? And then at a certain point, you only got 3. So I want to know, how did you face this situation or this point of your life as a researcher or as a scientist? Thank you.
I think I faced it with panic more than anything. I think I was very scared at that moment when everything stopped working. I think, you know, for researchers though, I mean, that actually is the #1 question that I get asked. I'm not sure about other folks in the audience, but that's the #1 question. You know, how do you deal with failure? And I think as scientists, we all deal with failure, and we're not great at talking about failure, but we all know we all sort of have to work our way through it. I'm a big fan of this very cliche statement that failure is just another word for experience. You learn from failure. You learn the things which do things, and you build up concepts and ideas from the failures. You start to realize that how more intricate and nuanced things are, and from that information you build. So failure is important. #1, it's important because it teaches you lots of things. It's also important because when you have success, you need the failure to really enjoy the success as well. But ultimately, I think all of us sort of believe in, you know, this idea of science. We can get to exciting new things. That's why we're all here, right? And so even when you're dealing with that failure or that lack of success, if you believe that this concept should work and there has to be a way to make it work, I think that in our inner selves provides motivation to just keep driving forward, keep driving forward. I think that's what pushes all of us.
Thank you.
Thank you, and I think this was a very inspirational Q&A session. Thank you so much.
Thanks.
For your time. Yeah, let's thank the speaker again.
Thank you, Sir David MacMillan and Prof Kozlov. All right, with this, we have come to the end of our morning session. Before we break for lunch, for those interested to find out more about what a marine scientist does, there will be a lunchtime online engagement by researchers from the St. John's Island National Marine Laboratory. You can join this lunchtime online engagement commencing shortly at 12:30 p.m. on the GAFNI platform. With that, I now invite you to join our speakers outside for lunch and further conversations. Please reconvene here by 1:25 p.m. for our afternoon session. All right, enjoy and see you soon.